participants to day 4 of the 5-day faculty development program on climate resilient agriculture and allied technologies for net zero and socially inclusive framework, organized by the MIT Institute of Organic Agriculture under the aegis of the Agriculture and Allied Sciences domain, MIT University, Noida, Uttar Pradesh. Over the past 3 days, we have explored global perspectives on climate resilient agriculture, climate informed soil, crop, water, and post harvest management, as well as the transformative role of precision agriculture, artificial intelligence, and digital technologies in sustainable farming systems. Today, we move into one of the most exciting frontiers of agriculture sciences, that is genomics and genome engineering for climate smart agriculture. The advances in genomics, molecular breeding, genome editing, and biotechnology are enabling scientists to develop crops that are more resilient to drought, heat, salinity, and emerging biotic stresses, while simultaneously enhancing nutritional quality and productivity. These innovations are expected to play a crucial role in ensuring food security under changing climatic conditions. So I would like to express my sincere gratitude to our visionary leadership for their unwavering support and encouragement. We are deeply thankful to our Honorable Founder President, Dr. Ashok K. Chauhan sir, Honorable Chancellor, Dr. Atul Chauhan sir, Honorable Vice Chancellor, Dr. Balwinder Shukla ma'am, Director General, MIT Food and Agriculture Foundation and Chancellor, Dr. Nutan Kaushik ma'am, and Director, MIT Institute of Organic Agriculture, Dr. Doily Sanyal ma'am, whose guidance and commitment have made this program possible. So with these words, let us begin day 4 of this faculty development program on the theme, recent advances in genomics and genome engineering for climate smart agriculture. With these words, I would now like to introduce our first distinguished resource person of the day, Dr. Rupesh Deshmukh. So Dr. Rupesh Deshmukh is an internationally acclaimed agricultural biotechnologist and genome scientist, currently serving as the professor at the Central University of Haryana. Dr. Deshmukh completed his PhD in agricultural biotechnology and subsequently established a distinguished international research career in plant molecular biology, genomics, and genome editing. He has held prestigious research and academic positions at university level, University of Missouri, and Plaxa University. His pioneering research on silicon uptake mechanisms in plant led to the identification of silicon transporting aquaporins across several plant species using advanced omics approaches. Currently, his research focuses on genome editing, crop improvement, stress resilience, and sustainable agriculture through biotechnological interventions. Dr. Deshmukh has received numerous prestigious honors, including the Ramalinga Swamy Fellowship Award, Dr. P.S. Khanakhoje Gold Medal, and NASI Scopus Young Scientist Award. His name has also been featured among Stanford University's World Top 2% Scientists for multiple years, with over 200 research publications and editorial responsibilities in several leading international journals. He continues to make outstanding contributions to agricultural biotechnology and plant science research. We are privileged to have him with us today. Sir, I respectfully invite you to deliver your lecture.
Thank you very much, Dr. Tarun, for such an extended introduction.
Welcome, sir.
And thank you for MIT for providing me this opportunity. So can I share my slide?
Yes, sir.
Sure.
Yes, sir. Please share and please start.
I hope my slide is visible.
It is visible, sir. Yeah.
Okay.
Visual, sir.
So basically, today's talk will be around aquaporin in different plants. Our group working with the aquaporin and silicon uptake, because many of the aquaporin, they are also involved in the silicon uptake. So we basically work with the most of the time the labs usually work with any specific plant species, But since our lab is more focused on the basic understanding of aquaporin and silicon uptake, we work with the several different plants, starting from the lower plants like alga and mosses, and then the angiosperms, gymnosperm, all kind of plants. We work to understand how this aquaporin evolved, how the specific aquaporin can transport specific solute. So this is our university. This is like almost 150 kilometers from Gurgaon, which is the NCR region. So we have very beautiful 500-acre of campus. And in one of this academic block, we have ground floor. So as I mentioned that our group basically work with the aquaporin, and we use different tools to better understand the aquaporins. And once we have the basic understanding, our group is also working to translate that basic understanding for the crop improvement programs. So in soybean, we work with the silicon transporter. Here we have identified the gene, and then we use genome editing tools to make the desirable modifications in the high yielding varieties. Then in case of mustard, we are working with stress resilience, since aquaporin are majorly water transporter proteins, and that has a major role in the stress. In tomato, we are working with aquaporin to improve the nutritional quality. And in case of rice, we are working with aquaporin to reduce the arsenic content. So basically, we are more exploring this aquaporin protein in rice, tomato, soybean, and mustard. These are the main four crops. And we also use diverse plant species to understand the evolutionary path of this proteins in higher plants. So basically, aquaporins are a channel forming protein. Those are made up of 6 transmembrane domains and 2 half transmembrane domains. Those form a constrict at the center of the aquaporin. So this is the membrane, cell membrane, and this aquaporin form a pore across the cell membrane. So whatever solute present outside the cell, extracellular region that can pass through this aquaporin pore and enter into cytoplasm. It will be reverse also, because and that aquaporins are also
Sir,
known to transport.
sorry to interrupt you. There are some
Yes.
problems in your slide. Some blocks are appearing, black color, some around your slide.
Okay.
You can please
Can
check.
I?
Some part of your slide is not visible.
So maybe this is because of this.
It's a design or slide design?
Now,
Uh, no, sir. Still, now it is better. Yeah, it is better. Yeah,
okay. Okay. So these aquaporins are known to transport glycerol, urea, ammonia, hydrogen peroxide, carbon dioxide, boric acid, and silicon. That is the area where we work. Silicon and arsenic, both are the metalloids. So many aquaporins, they also transport the metalloids. So this is the structure of aquaporin. They have 2 big opening. One is extracellular, another one is the intracellular. And at the center, they have 2 constricts, known as constrict formed with the selectivity filter amino acids. Of transmembrane domains, which has a NP motif. These 2 NP motif, they form the second constrict. So these 2 constricts are very important. They decide what type of solute can pass through the aquaporins. Aquaporins are big family. Like in case of soybean, there are 72 aquaporins. In case of canola, there are 120 aquaporins. But not all the aquaporin they transport water. Some of the aquaporin they transport water. Some aquaporin they transport silicon. Some transport boric acid. Some transport urea. Some transport carbon dioxide. So they evolve in a subfamilies, and
Sir, your voice is not audible.
these are the major. Yes, doctor. Is it visible now?
Now visible, but our video is not moving. Now it is fine. Internet. Okay, sir.
Okay, slide transition is slow.
Okay, sir.
Is it okay now?
Yes, sir. Yes, you can.
Okay, so we cloned these two genes, we characterized those, and we found that both genes they transport silicon, and these are the major genes in soybean, which help soybean plant to uptake silicon. And if soybean plant uptake more silicon, they will get a better resistance against abiotic and abiotic stress tolerance. That's why it is very important to identify the silicon transporter and use those in breeding high yielding varieties. So once when we clone these genes, we first transform those genes in Arabidopsis. We check the silicon uptake. Study the transporter protein for the solute transport and the transport kinetics. Okay, then we also cloned another silicon transporter that is from the mung bean. The difference between soybean transporter and mung bean transporter is that this has different selectivity build. As I mentioned earlier, in aquaporin there are two constrict. One constrict is formed with the 4 amino acid, and another constrict is formed with the 2 NK motifs. So the first constrict in soybean that has a GSGR, these 4 amino acids. But in case of mung bean, that is ASGR. That is the difference between soybean and mung bean. And when we characterize this mung bean silicon transporter using Xenopus oocyte assay, where we take frog eggs, then inject the cRNA of the aquaporin gene, and then we keep that oocyte in a bath of solute. And over the time we measure how much solute is transported into the oocyte. That's how this Xenopus oocyte assay works. And we found out that there is silicon transporter identified in mung bean is a active silicon transporter, and that has a significant role in uptake of silicon in mung bean. Okay. So with such experience of identifying silicon transporter and further characterizing the silicon, functionally characterizing the silicon transporter using Xenopus oocyte assay, using Arabidopsis as a transporter system, Arabidopsis transformation, and then yeast assay. These 3 assays we basically use to characterize the cloned aquaporin. Then we used whole genome sequencing information available in 1,000 plant species. This include very diverse plant species from the different uh, orders. So here we quantified silicon uptake in all these plant species in a same uh, setup. Like we grown those in a greenhouse, and we supplemented those plants with the silicon. So everything is the same. So this is from the single large experiment uh, uh, where we quantified the silicon in a 456 diverse plant species, and we find out that the species which are in the poles, they are a very good silicon accumulator, whereas species like brassicas, they don't accumulate uh, significant amount of silicon. And when we looked into the genome of these species, we identified that whatever plant species which has this silicon transporter aquaporin with a GSGR type of selectivity filter, they accumulate high amount of silicon. Whatever angiosperm, if they do not have a silicon transporter aquaporin with this GSGR type of selectivity filter, they are a poor silicon accumulator. From this large study, we identified that whatever species which has the silicon transporter aquaporin, those are high silicon accumulator. Whatever angiosperm or genomes are lacking this silicon transporter aquaporin, they are a poor silicon accumulator. So we can easily group plant species into 2 categories. One is a high silicon accumulator, another is a low silicon accumulator. And then when we look back into the evolution of the silicon transporter aquaporin, this is the evolutionary path. In alga, there is no silicon transporter aquaporin, and alga cannot accumulate the silicon. But in mosses, some of the mosses they have silicon transporter aquaporin, and there is evidence that the mosses can accumulate the silicon. Similarly, in club moss, gymnosperm, and angiosperm, many of the species they have silicon transporter aquaporin, then accumulate the silicon. When we look into this GSGR selective filter, over the evolution, this GSGR filter was evolved in a way that it become very conserved in angiosperm. Most of the silicon accumulating angiosperm they has this silicon transporter aquaporin with the GSGR selectivity filter. From this study, it is now confirmed that the silicon transporter aquaporin evolved 500 million year ago in a mosses, and now those we have in a angiosperms. Okay. So with this aquaporins identified in about 1,000 plant species, we started looking into conserved features, and we find out that there is a new conserved feature, that is the distance between two NPA domain. All the silicon accumulating plant species, they have aquaporin with GSGR filter, and in those aquaporin, the distance between two NPA domain is a 108 amino acid. So here you can see like Casanus casana that has a two silicon transporter aquaporin, both has 108 amino acid. Glycine max that has a 100 amino acid. All the species which are silicon accumulating and has this silicon transporter aquaporin, they have 108 amino acid spacing between two NPA domain. But we found two exceptions. One is a citrus senesis, where the distance between two NPA is a 43, and another one is a Solanum lycopodium, where the distance is 109. Only one extra amino acid is there. In rest of the species, there is a distance between two NPA is 108, and in case of tomato, the distance is 109 amino acid. So here it is very confirmed that the silicon accumulating plant species, they have silicon transporter aquaporin with the NPA spacing 108. Then when we looked into tomato and citrus, both found the poor silicon accumulator species. That means if the 108 spacing is not there, the plant species will be poor silicon accumulator. And to confirm this hypothesis, we cloned gene from populars, tomato, or rice. And for citrus, there is a big deletion. That's why it is we can easily imagine that why citrus are not good silicon accumulator species. But in case of tomato, there is only one massive difference. So we clone genes from this species, then we heterologously express those in a xenopus assay. Then we use each assay and we heterologously express those gene in Arabidopsis. So in oocyte assay, we found out that the silicon transporter aquaporin having 108 amino acid spacing from rice that show very high level of silicon transport activity. Same with the popular, which has 108 spacing. That also showed very high transport activity. But in case of tomato and citrus, both fail to show any silicon transport activity. In case of tomato, there is only one amino acid extra. Okay. Then to again reconfirm this, we use site-directed mutagenesis. Like here, in case of populus, the wild type has 108 amino acid spacing. Then we created mutant by removing glycine at position 139, and then the distance become 107. Then we added one extra amino acid at 139, that is glycine, to make a 109. Now we have three genes. The wild type, which is a good silicon transporter that has 108 amino acid. Then the mutant 107 by removing one amino acid. Then another mutant with 109 spacing, which created by adding one amino acid. When we tested all those three construct in a xenopus site, we find out that the wild type that showed very high level of silicon transport activity because that has a 108 amino acid spacing. If we removed one amino acid, that lost the functionality of this transporter. If we add extra amino acid, even though the functionality of this transporter get lost. So that means that 108 amino acid really play a significant role in a functionality of the silicon transporting aquaporins. Still, this is not very convincing result. Then we took a more challenging task, that converting the non-functional aquaporin into the functional, that is the tomato, which has 109 amino acid spacing. So we decided to remove one amino acid from this position to create a 108 amino acid spacing in a tomato. If our hypothesis is correct, then this evolutionary non-functional silicon transporter in tomato should convert into the functional one. Then we removed one amino acid valine from this loop, and then this become a functional. So this is wild type with 109 amino acid spacing, and once we removed one amino acid, that become 108 spacing, and that become a functional silicon transporter in tomato. And now we can say that these results are very convincing to uh, say that the 108 amino acid, which is very conserved across all silicon accumulating plant species, are really a requirement for this aquaporin to be a functional silicon transporter. Okay, and now question arises: if we improve silicon in a crop species, that will definitely help to improve the production. But what about the human health? Whether the silicon rich food is good for human or that is toxic for human? So there are several reviews you can see here that suggest that the silicon help to improve the bone health, to improve the skin health. So that is helpful for human consumption as well. There is one disease like known as a silicosis, but that is caused because of the inhaling silicon dust in the form of silicon dioxide. The silicon which we consume with the silicon-rich food is in the form of silicic acid. The body can uptake only in the form of silicic acid. Rest of the silicon in digestive tract, that will be excreted out, and that will not cause any disease. So here you can see there are several such articles are already published. And based on this research in a human, several silicon-based tonics are also in the market. And once these tonics are in the market, that means they have already conducted the trials and confirmed that the silicon is beneficial in not causing any disease. So all these silica-based tonics are already in the market. Okay, so moving on to the crops, more than 1,000 studies are there which shows that the silicon supplementation improve plant resilience under abiotic as well as biotic stress. So are there any option to improve silicon uptake in rice? Because rice is already a very good silicon accumulator, but if we improve silicon uptake, that may give us extra protection against the biotic and abiotic stress. There are more than 3,000 diverse rice varieties have whole genome sequence that are available. And when we looked into these resources, we find out that all the whole genome sequence rice variety, they have exactly the same sequence for the genes. So there is no genetic variation for silicon transporter lycopyrene in 4,700 rice varieties. These many rice varieties has a whole genome sequencing data publicly available. The gene is highly conserved. That means we cannot use conventional breeding to further improve silicon uptake in a rice. Okay, so what about the overexpression of the silicon transport? We can use a transgenic approach. We can just overexpress the well-characterized silicon transport in rice. But that is also not a good idea because silicon transporter also transport arsenic. And arsenic is a carcinogenic. So here you can see this is the India map. This many area of the India is arsenic contaminated. And this is the area of rice growing. So most of the rice is grown in this area. That means this is the major issue in rice that if we use the silicon transport, that will also uptake arsenic, and the food become contaminated with this arsenic element. There are already cancer trade running in Punjab, which is the rice growing area. So this is not a good idea. But if we knock out this silicon transport lycopyrene in rice, the name of this silicon transport lycopyrene is OS-nitrobus-1. So because of this gene, rice gets silicon as well as arsenic. And because of the arsenic contamination, the rice grain become toxic. Second option we have, we can just knock out this transporter to get rid of the arsenic. But if we knock out this transporter, that will also reduce the silicon uptake and because of that, there will be yield loss. So that is not the uh, approach we can use. But there is another opportunity where we can just modify this silicon transporter in a way that can allow silicon, but that can restrict arsenic. And we took this challenge and we got a project from CRA to test our hypothesis. So hypothesis is that the silicon transporter icoprene has this hourglass-like structure, and these two constructs are very important, which decide the solute specificity of the aquaporin. So this aquaporin form a pore. So we started studying the pore lining amino acids, which amino acid interact with the silicon and which one interact specifically with the arsenic. So we use molecular dynamic simulations uh, because silicon transporter aquaporin also has a high resolution crystallographic structure available. So we used that and we started studying the pore lining amino acids. And here one of the example, if we change this glycine at position 88 to valine, only single amino acid change can drastically change the entire pore. So this is wild type pore. This is big mouth outside the cell. This is big mouth inside the cell. These are the constricted. This is the pore diameter, the pore map. If we change only one amino acid, that can completely change the pore. So it is very critical to specifically make a change which can allow silicon transport but can restrict the arsenic transport. Therefore, it took more than 3 years to identify these specific amino acids. Once we identify these amino acids, like these are the pore lining amino acids, then we use side directed mutagenesis. And after hundreds of side directed mutagenesis, we identified these three amino acids. These three amino acids, where if we change those, like this is very important that Q at position 84, if we change to glycine, that will reduce 40% of the arsenic transport without affecting the silicon transport. And now my another student, Srija Sudatran, she is a NPDF in our lab. She is doing prime editing to change those amino acids in a rice grain using the prime editing approach. Okay, and we are the first to show that we can change the solute specificity of the aquaporin, and this provides us opportunity to also develop commercial product like the aquaporin based filter for the specific solutes. Okay, and in soybean to identify the silicon transporter, we used GWAS and QTL mapping approach, and once we identified the locus governing the high silicon uptake in price, like GWAS failed because very few soybean genotypes they accumulate high amount of silicon. Then we use QTL mapping. Here you can see in soybean we identified very large QTL having load score more than 40, and this is like a monogenic trait. And then we looked into this locus and identified silicon transporter genes in a soybean. And then actually in that QTL, there are three genes randomly duplicated. And to precisely understand which gene has what effect, we used a CRISPR-Cas based genome editing approach. And we created a catalog of soybean lines with the different combination of mutation in the homologs of this silicon transporter. We named the silicon transporter as high seed. And we use CRISPR-Cas based genome editing to knock out the genes. Okay, these are the deletion which we achieved in soybean with the genome editing, and then we studied the individual lines with the specific mutation combination to understand the silicon uptake. So in tomato, we used multiplex genome editing, where we targeted this SL-NIP2-1 silicon transporter. This is SP5G transcription factor for the plant architecture, then pectate lies. So these genes we targeted in tomato. And here you can see this SP5 gene that regulate plant architecture. Pectate lies that improve the shelf life of the tomato. Lycopene beta cyclase that improve the lycopene content, which gives better red color to the tomato. And this SL-NIP2-1, which is a silicon transporter in tomato, where we wanted to remove only one single amino acid so that the silicon transporter become the functional. Okay, these are the result in tomato. This is for SP5G. You can see this is Pusa ruby variety. If we knock out this SP5G, the plant become determinant, and the total fruit yield per plant will be almost same. The plant become more compact, and the cropping duration will be reduced. So we basically use 4 different genotypes to make this editing. One is a Pusa ruby, another is a Pusa early dot, then Pusa um, Solanum peruanum, that is the wild type species. So in all these different genotypes, we got a different type of phenotype. Like in case of Pusa ruby, wild type has this specific pattern: flower, first internode, second internode, third internode, and then flowering. When we make a editing of this SP5G, there is a flowering, then single internode, and then again flowering. The flowering become in every alternate internode. But in case of Pusa Lidop, which already have a flowering in alternate internode, after editing this gene, flowering become on every internode. So that is the genotypic difference which we observed with the editing of SP5G. These are the result from the editing of pectide lies. This one is a wild type, and this one is a edited one. So the edited one has almost 2 week extra shelf life, and that also reduced the fruit cracking also. This is the fruit cracking reduced because of the editing and pectide lies. Okay, and this is lycopene content in a edited plant. Almost twice lycopene is produced in the like one beta cycle is edited lines in tomato. Okay, that SP5G also improved the root architecture. This is the wild type, and in edited lines we found 3 times longer roots. This all work is from the tomato generating work is from the Sanskriti Vars PhD program. Now she is at the University of Copenhagen, Denmark. The molecular dynamic simulation of silicon transporter aquaporin in rice, that is from the Yogesh PhD work. He is doing PhD postdoc at Chinese Academy of Agriculture Sciences in China. Then Ruchi Bansal, who worked with the multi-omics and rice haplotyping, she is at the data scientist at Amazon. And then Surbi Kumawat, she worked with the aquaporin in fruit development in bottle guard. She is doing postdoc at Kansas State University now. Vandana Thakral, she worked with the Mungbean aquaporin. Result I have shown you. She is doing postdoc at Western Virginia State University. Gaurav Rotary, he worked with the silicon solubilizing bacteria, and we identified silicase enzymes in this specific bacteria. And he is doing postdoc at Texas Tech University. At present, Mukesh is doing PhD. He is in a 3rd year. He worked with the onion aquaporins. Badal is working with the multiplex genome editing in tomato. Pragati is working with the uncharacterized intrinsic protein. This is the one class of aquaporin. Pawan is working with the silicate solubilizing bacteria. Gitanjali is working with understanding role of aquaporin in metabolic transport. Akash is working on understanding aquaporin evolution specifically in the legumes. So thank you to our collaborators, students, and thank you to you for listening. Thank you very much.
Tarun.
Dr. Tarun. Yeah.
Dr. Tarun, yes, thank you, sir, for the excellent presentation. We shall now open the floor for interaction and questions.
Yeah,
Participants are, yes.
Audience have any questions? Yeah, they can. Professor,
Actually, I have one question actually.
Yeah.
So, sir, silicon you have recommended regarding there is arsenic uptake with the silicon application actually. There is option with the transporter wise of operations. However, we know silicon is recommended for the water stress management.
Yes, silicon is a beneficial for
Yes.
improving resilience under abiotic as well as biotic stress.
Yes.
Actually, silicon application reduce the arsenic.
Okay.
The transporter for the silicon is also transporter for arsenic. If we apply silicon to the arsenic contaminated field, there will be less uptake of arsenic since both are getting transported through the same transporter. So silicon will be beneficial for arsenic contaminated soil also.
So that's actually a negative thing with the silicon then.
No, it's not negative. If we apply silicon in arsenic contaminated field, arsenic uptake will reduce
Okay.
because the
Yes.
transporter involved in a silicon uptake and arsenic uptake is the same.
Okay.
And in our study, we are trying to provide a sustainable solution by changing that transporter in a way that can only transport silicon but not the arsenic.
Thank you, sir.
Correct.
Anyone else with the questions?
DK sir having
Yes,
question. Professor Porche having question.
First, Professor Enrico, then me.
Thank you. I was surprised that in more than 4,000 samples of
Yes.
rice, there was no variation at all. Have you any idea about what happens in the wild species?
Yes,
Because if that is the same on the wild species, would be a very interesting case.
Yes. We looked into 14 Oryza wild species, which has a whole genome sequence that available. And in those species also, the gene is very conserved. There are variations, but those are not non-synonymous. Those are synonymous variation. The final protein will be exactly same.
Now you are going to do some editing. What are the results in general terms, not specifically for the role of that gene, but as any consequence on the activation of different genes?
Okay, when we in the first phase of the project, we use site-directed mutagenesis just to make the desirable amino acid changes in this protein. And then we tested those edited, mutated genes to test how much silicon that is transporting or how much arsenic that is transporting. We use 3 assays. We make a heterologous expression in Arabidopsis, and we tested that in yeast, and then we tested that in a oocyte assay. In these studies, it looks like that if we change only one amino acid, that will reduce 40% arsenic transport without affecting the silicon uptake. And now we are using this information to make this precise one amino acid change in a rice using prime editing approach. So till date, we don't have those plant ready with us to go for the evaluation of arsenic and silicon uptake. But maybe in the next year, we will be having those results with us.
Thank you.
Yeah, Professor Rupesh, very great presentation. Am I audible to you?
Yes.
Yeah. For my knowledge, aquaporins, you are working in different plant system, monocot and dicot. Did you see aquaporins have conserved sequences all throughout in every monocot or dicot system? Or is there any variation in the sequences that you are working?
Actually, aquaporin is a big family that has a variation. And based on the variation, we further categorize aquaporin in subfamilies like NEEP, PEEP, TEEP, SEEPs. Those are the 5 major subfamilies. There is a variation across species, but in case of specific aquaporin, which has a specific solute specificity, they have some conserved features that confirm that solute specificity.
Okay.
And that's why it is very important to understand, identify these conserved features so that we can make modification in that to change the solute specificity in those aquaporins.
Okay. Alongside, somewhere you defined deletion of one amino acid in PAC somewhere in the slide. Does it reflect not only point mutation, rather have you seen somewhere frame shift mutations?
We specifically removed 3 nucleotides to just remove one amino acid to create a spacing of 108 amino acid spacing between NPM.
Okay. Thank you. Thank you, Professor.
There is a question from audience. Could you please read the question, Dr. Tharum, from audience?
Yes. So actually, there is a question regarding silicon-induced responses is helpful in bringing about multiple stress resistance.
Yes, true. Actually, silicon act as a moisturizing cream. In a general terminology, I can say that because we use moisturizing cream in a very hot climate as well as we use in a winter also. So what happens whenever plant uptake silicon, that go and deposit it in a apoplastic region, the area between two cells, and there that act as a like that capture the free water present in apoplastic region, so that whenever there is any insect attack, that act as a shield against that. When there is a drought condition, that act as a moisture reservoir. So in that way, silicon provide protection against biotic as well as abiotic stress, and the magnitude of the protection is very limited. It is not something like pesticide or resistance genes. This is very sudden, but definitely when we look into collective effect of the silicon supplementation, uh, that definitely provide significant uh, effect on improving crop resilience under abiotic as well as biotic stress.
Sir, in case of aquaculture, you are focused on, hello, sir?
Yes.
You
Yes,
have
sir.
focused on fruit development, but what about this salinity tolerance? If you add some other genes stacking together along with aquaporin, like high potassium transporter or other, we can increase salinity tolerance, no? I think.
Doctor, can you just repeat your question?
Apart from fruit development, aquaporin, we can also use this kind of genes along with high potassium transporter and other genes for stress resistance like salinity to increase the
Yes.
salinity tolerance. So did you consider to work on that aspect in future, sir?
Yes, many labs they are working on this aspect, like using silicon transporter based building to improve the salinity stress. And practically, as a fertilizer application, potassium silicate is also good uh, fertilizer application which can provide tolerance against the high salinity.
Yeah, because this is also
In Indian market, there are three types of silicon-based fertilizer: potassium silicate, calcium silicate, and sodium silicate. So sodium, like potassium silicate, that has a dual benefit. Like potassium is also a major nutrient.
yeah.
That's why it's preferred to use a potassium silicate over the sodium silicate.
Yeah, because some of the genes like high potassium transporter, they are playing a major role. So we add some more genes, maybe it can have a more resistant to salinity as well. Suggestion. Audience, do you have any questions from audience side?
Okay, Tarun, please go ahead.
Next speaker is waiting. So,
Okay. So thank you very much, sir, for your
Sure.
insightful and enriching presentation.
Thank you, sir.
Your
Thank you.
valuable expertise and thought-provoking deliberations have significantly enhanced our understanding of the topic.
Thank you.
Thank you, sir.
Thank you so much, sir.
So it's our time to introduce our next speaker, Dr. Riazul Rauf Mir. So Dr. Mir is currently serving as the Associate Professor and Researcher at the Murdoch University, working with the Centre for Crop and Food Innovation at the Western Australia State Agriculture Biotechnology Centre. He also maintains strong academic and research collaborations with the Sher-e-Kashmir University of Agriculture Sciences and Technology of Kashmir. With over a decade of research experience, his work focuses on genome-assisted breeding, crop biotechnology, climatic resilience, and nutritional enhancement of crops. His research addresses critical challenges such as heat, drought tolerance in wheat, biofortification of legumes with zinc, iron, and protein. Being a gold medalist during his postgraduate studies and a recipient of prestigious recognitions such as DVT Crest Award and Young Scientist Award, Dr. Mir has made significant contributions toward climate resilient and nutrient-rich crop varieties. His interdisciplinary research integrating genomics, molecular breeding, and biotechnology continues to advance sustainable agriculture and global food security. Sir, we are delighted to welcome him today to today's program. The platform is all yours, sir. Dr. Mir?
Hello. Yes.
Hello, sir.
Thank you very much for your nice introduction. Yeah, it is always pleasure to connect with people back home in India. So, am I audible?
Yes, sir.
Yes, sir. Yes, sir. You are audible.
Yeah, first
Your
of
slide
all,
is
I would like
visible.
to thank you, my dear friend Karthikeyan, who was known to me for the last several years, and we have been meeting
Thank you, sir.
at different forums. So it is always nice to meet him again today through the Zoom meeting. So yeah, thank you all other organizers also for arranging this presentation. So it's nice, like I will be
Thank you,
interacting
sir.
with you guys.
Really, your lecture
So
will be valuable.
this, although the topic was different, but I then requested that let us present the work which we are currently doing here at Murdoch University. That's why I changed this topic a little bit, like which is enhancing nitrogen use efficiency and resilience to heat and drought in Australian wheat. So what I will be discussing in this presentation, as you can see in this slide, so I put three different aspects. Number one is nitrogen use efficiency. Number two is heat tolerance. Number three is drought tolerance. All the three are very important in Australian context. If you see, Australian agriculture is usually rain-fed. So heat and drought are two important aspects of wheat research. And the nitrogen use efficiency, nowadays it is becoming also a very hot topic in view of this this Middle East crisis. So these are some of the things what we are doing in nitrogen use efficiency. I have summarized it here in this slide. You can see like we have different mapping population in the center of this. And then what we are doing field phenotyping. And then we are trying to evaluate the effect of those genes which we have in these lines in the field. And then we are doing some physiology, we are doing proteomics metabolomes in this project, and then we are also validating some of the genes through genome editing. And then, of course, we are mostly doing genomics, so a lot of sequencing work is happening in this project. So in the next project on heat tolerance, so again, we have this diverse germplasm collection. We are conducting field trials and also trials under the controlled environment in greenhouse. Then we are sequencing some of those collections and we are identifying genes and haplotypes. So in another project which is on the drought, in this case we have some diverse germplasm and we are conducting field trials, in this case also in different locations in Australia under rain-fed conditions. Then we are sequencing some of the germplasm, and based on the sequencing we identified some of the important genotypes. And those genotypes we are developing pangenome and haplotype catalog. So these are some of the important projects which I will be discussing in my presentation. So moving to the first topic, which is nitrogen efficiency. As you know, farmers, particularly in India, not in these countries where everything is calculated, particularly in India, we use excessive nitrogen in agriculture. So when you use excessive nitrogen, but we know the use of nitrogen not only in wheat but different crops is inefficient. Like in wheat, it is only 40% of the applied nitrogen is taken up, like 60% is not taken up by the plant. And among that 40%, only 33 ends up in the grain. This means more than 67% is wasted. So excessive use of nitrogen, it is bringing up a lot of impact on the environment by causing nitrogen pollution and the concerns to human health therefore. So when you apply heavy nitrogen to the agriculture, particularly wheat, we have increase in internode or basal internode length. This results in the lodging, as well as when there is lodging, sometimes there can be 100% yield loss. So moving, what is nitrogen and how is nitrogen use efficiency being calculated and what are its different components? You can see in this case, nitrogen is very important, like it is part of plant hormones, pigments, chlorophyll, nucleic acids, amino acids, proteins. So this means it is very important for growth and yield of particularly wheat and all other cereals. And moving to the different components of the nitrogen use efficiency, you can see nitrogen use efficiency, we have two important components: uptake efficiency, which is actually the ability of plant to remove nitrogen from the soil. Then we have nitrogen utilization efficiency, which is actually the ability of the plant to use nitrogen to produce grain yield. This is very important for us, for breeders and for farmers also. So these are two important components. I will be dealing with this in some of our slides. So then how is nitrogen efficiency important? You can see what is it is doing. We can reduce the nitrogen losses. We can minimize greenhouse gas emission. we can prevent soil degradation, lower fertilizer costs, increase farmers' income. We can enhance crop yield, and then how we are there are different ways to calculate nitrogen efficiency. How we are calculating in nitrogen efficiency, you can see we multiply grain yield into grain protein content. These are two important components in our research trials. So we often do this, like we have to measure yield and protein content, then we divide by this to soil nitrogen, because we often measure how much nitrogen we are applying. So what is like, you can see the low nitrogen efficiency and protein, grain protein content. These are the historical problems in Australian agriculture also, in particularly wheat production. Why? Because soil type in Australia is often limited nitrogen availability and supply to plants. Why? Because the rain pattern in Australia is we have either the more rain. If you have more rain, then you have leaching of those nitrogen fertilizers. But if you have less rain, then the uptake is very difficult for the plant. The another problem is when we have more yield, then the protein gets diluted, because there is a famous protein dilution effect. When you want to increase the yield, protein dilutes. So there is a negative trade-off between protein as well as yield. So this means there is always this negative relationship happening. This is not happening only in Australia, this is happening everywhere in the world. And the challenge for improving, therefore, this nitrogen efficiency is, like, nitrogen efficiency versus grain yield and protein content. There is always a tug of war, which we call in breeding terms, there is a linkage drag. So high nitrogen efficiency means plants are very efficient. They use the nitrogen for efficiently and making higher yields, which is great for all of us. We want that. But however, when we increase the yield, it dilutes the protein. I will show some of the examples how it dilutes the protein. So it is called nitrogen dilution effect. So what is the solution? What is the way forward? What we are doing? We are doing some of this MABC, marker-assisted backcrossing approaches, using to break this linkage, break this undesirable linkage. And that is one of the challenges, and that is one of the solutions that we can do to alleviate this problem. So, but again, this nitrogen use efficiency, this is not a simple trait like we have one or two genes, but this is a very complex trait. Complex trait means you have large number of genes, and these genes are interacting with each other in different combinations. And this means it's a very complex quantitative trait influenced by the environment also. And wheat itself is a very complex genome, where you have 3 different genomes, A, B, D, and that adds to complexity of improving this nitrogen efficiency. So moving to the states, if you check the states of wheat area, then the nitrogen, how much nitrogen we are applying into the agriculture, particularly wheat in India, in Australia, and then how much cost. Like in Australia, we are nitrogen cost, it accounts to $556 million. And if we see this only in Western Australia, which is the biggest state, $180 million is only the cost for the nitrogen. So if we want to improve the nitrogen efficiency, like maybe 5%, 10%, 15%, like if we calculated 10% improve in the nitrogen efficiency, this means you can save $55.67 billion in whole Australia, and $18 million in Western Australia. So what are our approach, how we approach it, what we are doing at Murdoch University? You can say, like, the first thing we did QTL analysis and discovered some of those promising QTL clusters. So how we discovered those QTL cluster, you can see this in this, like we have initially selected 6 different populations. These are the biparent populations. They were developed by crossing like one parent with another parent, because that's why we call them biparent populations. So we had the 6 biparent populations. How these parents were selected, you can say some of those parents were high grain protein content parents, like Spitfire is from Australia. And then Bethlehem is from the Israel. This is the name of the place in Israel. You might be hearing about this place nowadays in the news. Where the conflict is also happening. So Bethlehem is a landrace from Israel, high protein line. Similarly, we have high yielding varieties from Australia like maize, Westonia, and Suntop. They were used together, and these 6 populations were developed. So they are all the double haploid populations. And then we have grown those double haploid populations. You can see in this map, there are like 3 locations in Western Australia, Morassic, Beverly, and Williams. and one location in Narrabri. This is called the wheat belt of Australia. Like towards the edge of this map, you can see this all is the wheat belt of India, Australia. So we selected 4 locations for 2 consecutive years. This is how phenotyping was done at these locations. So in addition to phenotyping at different locations, what we did, these 6 populations were genotyped, used 90,000 SNP markers. And using this phenotypic data as well as this SNP data, 6 individual genetic maps were developed for 6 populations. As I told, these were the 6 populations. But then we merged all those 6 populations together and developed a single consensus map. This is the consensus map of the 6 populations. And if someone is interested to know the details of those 6 populations, genetic map of 6 populations, here is the detail in this table. You can see like this is the population name, then the parents which were crossed, then the population size and the number of SNPs on each map. And using this genotypic data as well as phenotypic data from 4 locations on nitrogen efficiency traits. So what we did, we identified QTLs and genes. You can say genes per grain protein content, grain yield, nitrogen efficiency. These are the 3 important traits which we are using for calculating this nitrogen efficiency. So as you can see this slide, we identified those QTL genes for grain protein content, grain yield, nitrogen efficiency. In total, 156 genes were identified from all those populations. And now you know more than 150 QTLs. It is not easy to work with. Breeder always get confused. So what can I do with these many QTLs? This means you have to come up with only selected QTLs, very important, which breeder can work. So what we did, we selected those important QTL clusters. So we selected total 6 QTL clusters. In this case, this was the criteria how we selected, like stability. We want to select the QTLs which are controlling all the 3 traits. Like in this circle diagram, you can see the inner one is the grain yield, then this is the grain protein content, nitrogen efficiency. Like here is a cluster 1 which controls all the 3 traits. It is on 2B, then we have another on 2D, again controlling all the 3 traits. Then we have another one as 4D. It is also controlling all the three traits, yield, protein, as well as nitrogen efficiency. Like here, another one is controlling only 2, grain yield and grain protein content. So likewise, this was the criteria. Select genes which are responsible for all the 3 traits, yield, protein, as well as nitrogen efficiency. And select the genes which have a higher effect, like which contribute higher phenotypic variation. And select the genes which are detected across all the 6 populations, or if not 6, then maximum number of populations, like 4 population, 3 population, not in a single population. So this was the criteria, and this means using this criteria, we narrowed down only 6 important clusters. And what we did from each 6 QTL cluster, in order to validate those genes in the field, because you give these genes to breeder, they will again say, have you validated them before providing us for further improvement? So we wanted to validate those genes. As you know, this is the details of those 6 genes clusters, and now different environments where the genes have been identified. I mentioned some of those, and this means I am not going to tell more details about these clusters now. But what we did in the next step, when we have those 6 clusters in this, like cluster 1 to cluster 6, what we did, based on the allele present on those population, we selected these positive lines, like we have the positive effect of the QTL, versus negative lines where we have the negative effect of the QTL. That is how you can validate them in the field. We try to put both lines in the field. See if we have the positive effect of the QTL, whether it is really translating into performance in the field. If it's not translating, then you can say, oh, this QTL is not valid. So this means for all the 6 clusters, this thing was done, like we identified positive versus negative lines. So the first step was we wanted to validate them in the field. This is one of the pictures where we put them in the field, and this shows the contrast of those different lines, as you can see from this picture also. So in this figure, I want to say when we conducted multi-location trials, so these were the different traits which we targeted, although several other traits were recorded, but I want to give emphasis on these 3 traits. Then we did this validation in 2024, 25, as well as in 26. This year is also happening. So 3 years to confirm. And to confirm this, we added like N100 means this is the optimum nitrogen concentration. Here in India, we sometimes go up to 120 kg per hectare, but here 100 is considered optimum. And then N50, which is half nitrogen, which is the nitrogen stress. So when we put those lines under nitrogen stress, you want to test the performance. So we give the optimum level versus half nitrogen. Then you want to test them in the field, and these are the different locations. You can see we have some location in Western Australia, some in South Australia, and some are in the New South Wales. So 3 different states have been targeted. These are considered the wheat belt areas, like hard zones for wheat cultivation. Again, this is one of the screenshots of the trials in Western Australia, and this is when we visited the trial at harvesting stage. And I am just highlighting some of the results of 2024. This shows like for nitrogen efficiency at one location, Nardam, Roseworthy, this is South Australia, this is NSW. You can see on the left side, we have a negative line here. On the right side, you have a positive line. Those the lines where you have the positive allele, they always show the higher mean for nitrogen efficiency than the lines with negative. Again, you have here again at other location again showing positive lines performing better. Third location again showing positive lines performing better. This gave us confidence. Yes, our genes which are in those lines, they are performing better than the lines where we have the negative allele for the genes. Similar thing is happening for protein. You have the improvement of protein across all the 3 locations, like this is the Western Australia, South Australia, as well as NSW. And same thing is happening for the yield. This provides us the initial clues. Yes, the genes are working and we are able to validate them in the field, but this was 1 year data. And moving to see the another population, as I told, we had 6 populations, so this was the huge experiment. In the second population also, you can say like nitrogen efficiency is enhanced in positive lines when you compare with the negative lines. Then protein got enhanced, similarly yield got enhanced in the all the 3 locations. So this was the example for another location. So same thing we repeat in 2025. This is some of the results I will show for the 2025 also. So this is how we constitute those trials. Emphasis being put on the layout. You can see in this layout, we want to make sure like each genotype is repeated at minimum 2 times, but sometimes it ranges to 4 to 6 times. You have those replications. Then once we have those replications, then we have those different nitrogen levels. This dark patch, like they shows like we have high nitrogen concentration, which is optimal for us, and those light patches shows these are the genotypes where you have half of the nitrogen. So again, this is NUA and NUB. These are the different trials at one of the locations. Similarly, we have to design the experiment for all the other locations. These are some of the pictures when we visit those trials to see the performance of those genotypes in the field. This is the beginning and this is at the flowering stage. And these are again some of the screenshots. You can see these are the huge like trials where you have more than like 1200 plots in a single location where you have the genotypes under low nitrogen versus optimum nitrogen, then different replications also. And if you check the which the NDVI values, like using drone phenotyping, you can see like in the initial stage, like when the plant were at the vegetative stage, flowering has not started, you can see highest values of NDVI and they goes on down, like, you know, with the crop progression, NDVI value decreases. This is true. And then we have 2 nitrogen lines, N1 always more nitrogen, which is optimum, then N2 you have low nitrogen. So this is evident from this NDVI like drone phenotyping. Also, you can see the nitrogen stress was visible in the field in all the population. You see N2 low values. This means this was the nitrogen stress, and you can see this was successfully reflected in the crop canopies also in the field. And if I summarize those results of 2025 in short, you can see like this is N1 is the optimum nitrogen, N2 is the nitrogen stress. You can see there is not much difference, significant difference in N versus N2. This means under nitrogen stress, the genotypes where we have those genes, they are performing better. So your yield is not decreasing, but you see the protein and yield always there is a trade-off. At N2, when you are reducing this nitrogen, the protein is also decreasing. Although yield is not decreasing, but the protein is decreasing under nitrogen stress. This shows the nitrogen is very important for this protein. But when you see the nitrogen use efficiency, since at N2 we are putting half nitrogen, that is how nitrogen use efficiency is increasing here. because this shows the effect of genes. And when we plot all the three traits together, yield, protein, as well as this nitrogen efficiency, and if you see, you want to see the number of lines which perform better, like we have seen, like 12 is the standard protein content in wheat, 12, 12.5 sometimes. And if we have seen like after 12.7 protein, then more than 4 tons yield per hectare. There are several lines which can like show the trade-off doesn't exist. This means these lines have both high protein as well as high yield. That's what we are looking for. Among like 600 lines, we have some lines like 10, 20 lines. We want to shortlist 10 top 10 lines which don't show this trade-off. That is very important for us. So this was one of the approach. We want to validate those genes in the field, but then another approach is we want to validate through sequencing of those QTL clusters. So how we did, like we had those QTL cluster, US QTL cluster 1, 2, 3, 4, 5, 6. And then we had constituted those different lines. We have some positive lines, some negative lines. So we selected equal lines, equal positive, equal negative. You sequenced them. And we sequenced them like up to 20x coverage, like more than 300 gigabase data was generated for each pool. And this the pool, as I told, pool was selected. We have a positive pool as well as negative pool. But if you are using phenotypic data, we have to use the pool where you have less protein versus high protein, less nitrogen use efficiency, high protein use net high use efficiency, or less grain yield versus high grain yield. So we select those bulks, low bulk versus high bulk. We sequence them to 20x coverage, like more than 300 GB data was generated. And when you generate the data, then we have the QTL-seq pipeline. And we selected, we identified back those QTLs, which we have identified. So after QTL sequencing, this is the fastest method to identify the genes. Those sequencing pools like resulted in identifying back those QTL clusters which we have identified. And within each cluster, we can identify several candidate genes like cluster 1, 5 genes. So we shortlisted 5 genes from each, and now we are validating from them through qPCR. And one of the QTL cluster, I want to show you 2B QTL cluster, which we validated in the field, but through QTL sequencing, you can see this cluster was like in the genetic map. This was like from spanning from 54 MB to 72 MB. Through QTL-6, we can find exactly the same region here. So through QTL-6, you can hit the same region. This means you can say that we validated this region. But the other important feature of this QTL-6 is like the same region, like the more than 20 MB region, 18 MB region. We narrow down this region to 2 MBs, like the same QTL has been now narrowed down to 2 MB region. So we can say through QTL sequencing, we have done fine mapping of one of the region also. So this is very important. This in future will help us to clone the gene. So this is the another region where on the chromosome 2D, where we have this region spanning between 604 to 620 MB, like near the 16 MB region. So we could see like the same, like a little bit difference through QTL-6. So this means we validated this region. In addition, we identified several other candidate genes which are being validated. So this is not the story. In this project, we have also a physiology and biochemistry component. So in physiology and biochemistry, what we are doing, we know there are several genes or enzymes involved in nitrogen uptake, assimilation, and then remobilization into zinc, like which is seed in this case. So we targeted those components like enzymes which are involved in uptake, assimilation at different components, and then we quantified them. I will show some of those results. So in this slide, as I told you, like if you can say on this left panel, you can see on the top upper quadrant, you can say like we have those lines where you have both higher leaf nitrogen as well as higher grain nitrogen. What does this indicate? That indicate us these are the lines where you have more nitrogen in the leaf as well as in the grain. This means these are efficient lines where the translocation is happening like very efficiently from leaf to grain, as well as they are maintaining like high nitrogen in grain as well as in leaf. So these are the best lines which we selected like among the 600 lines. On the right, this panel, you can see these lines, they're in the lower right quadrant. You can see these have the lines which have low nitrogen in the leaf but high in the grain, because in this study we targeted leaf nitrogen not in leaf only but leaf as well as grain also. So we are trying to quantify the nitrogen in both together. So you can see in this panel it was high in leaf as well as grain, but here you can see it was high in grain but low in leaf. So what does that mean? In those genotypes, more and more nitrogen gets translocated into the grain. So these are also, this is the desirable for us, because at the end we don't want much to be in the leaf. We want it to get translocated, remobilize it into the grain. That is the important for us. So some of the genotypes, like on this lower bottom, you can say these were the efficient genotypes. So now then we have the physiology components. In physiology team, what they are doing at deeper, they are putting like 4 different nitrogen concentrations in different plots, like 10, 50, 100, 150 kg, to optimize the nitrogen concentration. In this case, so what they have done, they have selected top winners, like for nitrogen use efficiency, nitrogen uptake efficiency. So these are the important genotypes which have all the like very important for uptake as well as utilization efficiency. So these are some of the physiology which has been done. So finally, as a breeder or someone who wants to have the varieties for the growers, so the final component is, when we have the genes, we validated them in the field through physiology, through biochemistry. You want to introgress, transfer the genes into some Australian background. So we did this in 5 elite Australian varieties through MABC approach. So this is the pipeline which we are using. Like we have the QTL crosses, we have those we make crosses. We have the QTL like cluster on 2B and 2D. As I told you, in 2B we had 5 genes either within this region. In 2D we had the 2 genes. Then we developed F1s, the normal back crossing pipeline. Then BC1 F1s, BC2 F2s, BC2 F2s, and BC3 F1s. And this is an ongoing program. And in this slide, I want to just show these the names of the 5 important varieties which were targeted, like the Ironbark, Leverage, Shotgun, Sunmaster, Sundancer. These are the 5 you can say promising varieties which are currently being grown in different states in Australia. So they were already so high yielding, having good disease package and quality package. So what we did, we used those donor genotypes which we identified, where we have those genes which we validated and we know they were the important genes. So we just made some crosses and transfer those genes, and this shows the highlights some of the F1 spikes in different crosses, the number of F1 seeds we produced in each combination. So efforts were made to make sufficient F1 seed initially, and later on each backcrossing, sufficient backcrossing was done. So for backcrossing, as you know, these are 2 important components. You have to select the gene, track the gene, whether, because you don't want to lose the gene in different generations. So we did foreground selection using 21 SNPs using Intertech platform. So this is the Intertech platform which we were using for this foreground selection. Then we have this AgriSeq 5K, 5,000 SNPs were in AgriK thermofisher platform. This was used for background selection in all generations. If you see the details in this 5,000 SNPs, you can see the distribution of those SNPs. They were distributed on all the different chromosomes. And these the different SNPs were, they were like earlier, like the platform was built like you have the SNPs which are responsible for different agronomic traits, some disease resistance, physiology, quality, and different things. So all those genes have been put in the SNP, and the SNP is being used for background selection. So in this slide, I just want to show some of the screenshots, because we had 13 of those combinations. It is very difficult to show all of them. Just showing the screen, one of the slides where we shows like for foreground selection, like in this case, among the 100 lines, we tested 46 lines. And among the 46, these are the some of the lines where we have all these SNPs present, which are our target SNPs. And then this is the background, like all this red and blue spot shows this is the background in those lines, except those blue dot shows this is the donor segment which we are trying to remove in each generation. So this was the after backcross second. So we reconstituted more than 90 in some of the cases. So that is desirable. If we reach more than 90% recovery, that is what we want actually in this case. So this is how we used this AgriSeq platform to do background selection in each generation to reconstitute the recipient parent genome, so that we have those genotypes, new genotypes where we have the gene and we have the sufficient recovery. And in summary, in this program, I want to summarize this, like we have those 6 QTL clusters, cluster 1, cluster 2, and we have choose those 5 Australian recipient genotypes, which are very popular across Australia. Then we selected these donor genotypes for cluster 1, 2 donors, cluster 2, again 2 donors. And these are some of the features of those donor genotypes. Like you can see protein content more than 12%, then yield is also more than 4 tons. This is the scheme we use it, crossed those, then F1, again, back crossed BC1 F1. Then we have selected those 4 count background selection, selected few individuals, back crossed them, and each back cross, again, you have to cross back. So right now we are in BC3 stage and BC2 F2 stage, and these genotypes have been now in summary. So we have 18,000 plants now growing in the field, 18,000. So among the 18,000, we need to make selections, and now in next generation, we are now trying to progress only maybe 1,000 lines finally in the field. But right now we have more than 18,000 delivered to the industry partners, where among those 18,000 thousands will be tested in coming generation. So moving to the another project, I will not go through more in detail, but I hope I have some time left. So this is on heat tolerance. So this was some of the recent publication, if someone is interested to go through detail and what we are doing in this. So in this case, we are doing physiology and genomics perspective in this case, and different components, as I told you earlier. So we had tested 345 genotypes in the glasshouse. You can see like when we grow them genotypes in the glasshouse. And then at flowering stage, you transfer those plants at flowering stage to heat chamber. So in heat chamber, we maintain the temperature more than 38 for 5 to 6 consecutive days. So at the flowering stage, move the plants from this control condition to heat chamber, put them there for 5 to 6 days at high temperature, so that we want to see the effect of the heat on the seed setting. So this is some of the screenshots where you can see like this is the line before heat stress, this is the after heat stress. But when you bring it back after 5-6 days, you can see how the plant is recovering, and this shows the difference between one heat stress versus heat tolerant genotype. And this was how we were doing this heat screening under control condition. But at the same time, we are doing this in the field also. You put the collection in the field, and in field, it is very important in order to expose the crop heat stress. We have to do the 2 type of sowing. Sowing 1 is timely sown, but after maybe we delay another 1 month so that the crop gets exposed to heat stress at the time of maturity or at the time of flowering. That is the important critical stage for heat stress. So this slide shows like we have the same trial at 2 different dates of the sowing, and that was done. So in summary, if you can say, we had these 345 genotypes in the here. So what we did phenotyping under control condition, timely sown trial, and the late sown in the field. And field, often we use drones to capture different indices which are very important for measuring heat tolerance. Then in glasshouse, you have also in glasshouse no stress under normal condition. Then we exposed them in the heat chamber to 36 and 38 degree for 5 to 6 days sometimes. And what are the major findings for this case? That if you know the chlorophyll content gets reduced under heat stress to 33%, leaf area 21%, grain weight gets reduced to 51% under heat stress, then 1000 grain weight 33%. This all is like in this published results now. So overall, in heat, sensitive lines show the loss of chlorophyll, poor seed setting, accelerated senescence. So the key discovery in this is like higher chlorophyll retention, stable. Those heat tolerant genotypes, they retain chlorophyll, they are stable, they are better grain number and 1000 grain weight, and lower yield penalties in those tolerant genotypes. So in addition to this phenotyping, we did genotyping, SNP genotyping, then conducted structure, diversity, etc. And this is PCA plots. All of you may be knowing this. Then we conducted genome-wide assessment studies using different SNPs, and we had like 63 traits in this case. So 123 MTAs were detected, and the main highlight of this is we have some of the haplotypes where we have the same haplotype which responds for leaf area, grain weight, and for heat and drought. So the same region is controlling leaf area, chlorophyll content, and heat and drought. So these were some of the highlights of this study, and this has been published recently. So in addition to this, we have another project where we are targeting drought. Drought is very important under custodial conditions. So we have 300 diverse genotypes. We subject it to 3 different locations. One is Lagrease. Lagrease is the Western Australia. Then we have Pinery in Adelaide, South Australia. Then we have Pineru, which is towards the Victoria border, but in South Australia only. So 3 different locations. These are the rain-fed locations, like these locations where you have got less rainfall. This is the PhD project of Miss Anu Chettinini. She is spending a lot of time in this project. So in this case, you can see like the grain yield for the during first year, you can see like the grain yield at Lagrease, Pinery, Pineru. You can see Lagrease and this Pineru location. This was the heat stressed location, drought stressed location. Was less grain yield, but see the grain yield is less here. The protein is high. Because when yield reduces below a threshold, this means protein increases. That is the negative trade-off. Yield less here in Pineru, but protein is high. If you see here, yield is high, protein is also high. This is the favorable location for wheat, Pinery, South Australia, but here protein yield is more, protein is less. So these are some of the important correlations, but these are known also, like negative trade-off between grain yield and protein. But here we want to show the effect of drought stress on these traits. So in addition to measuring these drought at different locations, what we did, we sequenced this collection up to 12x coverage, and then you can see using different those phylogeny diversity, and as you can classify genotypes at different countries, as well as whether they are the breeding material, that is the breeding material, land race, advanced breeding lines, release varieties, etc. So you can see a clear distinguishing in this dendrogram. And based on this clustering, we selected 11 diverse genotypes. These highlighted genotypes are the 11 diverse genotypes, and they were sequenced, deep sequenced using PacBio and OmniC platforms. And then these genotypes represent more than 90% of the diversity of the bigger collection, 300 lines. Then we developed genome assemblies of all the 11 genotypes. And then we are checking the synteny, etc. The aim is to develop a pan genome. The pan genome will be used to call synapse structure variants, which will be used for working out genoid studies as a haplotype spar. Yield under rainfall conditions, or you can say drought conditions. So I would like to thank all colleagues. Like this program is managed by on the top by Professor Varshni Anu Chettinini, who is leading all genotyping efforts. Then some senior colleagues are there, Vanika Garg, Junjuan, Vikas, and some PhD scholars are involved in this work, and several colleagues who are part of this project. So I would like to thank all of them, and this is the project team involved. So thank you very much for providing your attention. I hope I am on time. I don't know, but maybe I took more time.
Thank you, sir. Thank you very much for your impressive lecture. Dr. Tarun, let us ask audience
Yes,
if they have any questions.
thank you, sir, for the excellent presentation. We shall now open the floor for interaction and questions. Participants are requested to kindly raise their hands or post their questions in the chat box.
Yeah, so even if
already
someone
one
cannot
question.
raise the question, so you can provide email, they can later on also write, so no problem.
Yes, sir. Yes. Sir, actually, I have
The genomic stuff to identify
Yes.
genes, or maybe to find out the effect of genes in the field. So in this case, our aim was to identify like genotypes which maintain higher yield as well as higher protein.
Yes.
Not at one location, across the location, like 5
Yeah,
locations.
so
in this
there
case.
is a heritability also, good heritability among your
Good
genome.
heritability, yes.
Okay.
Heritability was good because we grow those genotypes in the wheat belt zones only, like there was no stress location for this case. But for drought, yes, you have to put those genotypes under drought stress, the location where you don't have the sufficient rainfall, etc.
Yes, yes. Sir, are you trying to find out candidate genes associated with this kind of?
Yeah, in this case, actually, this was the first project where genes were identified for nitrogen deficiency. In the second phase, they are being validated. But in addition to that, when we did the SQL sequencing using those pools, like high and low pool, we identified candidate genes for those different traits. And finally, we shortlisted 4 to 5 candidate genes for, again, validation through qPCR. I have not gone through the genome editing component. So
Yes.
we are validating some of those genes through genome editing, and we have edits. And then finally, they will be tested at different nitrogen regimes, like no nitrogen versus high nitrogen. If we find them the effect at the good thing in our case is we have genotypes which perform better even at low nitrogen. So when you have nitrogen citrus, this could be like when you have more
Like
nitrogen, plant have more
phosphate
dictated growth.
starvation tolerance, PUP1 locus, perhaps
Yeah,
there
yeah.
may be something for nitrogen also.
Yeah, so in this case, we are only trying to find out the validation of our 4 or 5, 6 genes, but there are several genes which are already known.
Yes, yes.
So
Key
our
genes, you
aim
are
is
trying to find out some more key genes, I think.
yeah, there are several other genes identified different groups.
Hmm.
So they are there, but yeah, we are trying to find out like that. In this case, we have those DH populations because we know the DH population, which allele, which QTL is there.
Yes, yes, sir. Sir, Professor Porcedu from Australia, sorry, Italy, is
Yeah.
trying to ask some questions. Sir, Professor
Okay.
Porcedu, please ask your question.
Thank you. Thank you and congratulations for your presentation and data you have. My point is whether you have any information on the distribution of the additional nitrogen to different protein components and their effects on the quality of bread and quality of biscuits.
Yeah, thank you very much. This is very interesting, but we have not gone through that much detail because what we are doing, we are just measuring protein content, not going into the fractions, quality, which fraction, bread making, etc. So no, we are not dealing with that component. But maybe in future, once we have narrowed down those best lines, like 10, 20 lines, which are best performing across all the locations, maybe those selected lines will go for further quality testing, not now. Because right now we have a big pool of 600 lines, 5 locations, different nitrogen regimes. So it is difficult to test these traits like chapati, bread making, or gluten, or different fractions. So maybe in future when we are narrowed down for finally 4 or 5 or 10 lines, maybe then it will go to more detailed phenotyping.
Okay, sir. Thank you. DK, sir, you have question?
Yeah, Dr. Rayaj, nice presentation. Many congratulations.
Thank you, sir.
Sir, being a microbiologist, I was roaming around to find out a good, suitable question for you.
Yes, sir.
Yeah. Sir, you have discussed that excessive level of nitrogen may cause lodging in the wheat.
Yes, sir.
Do we make some efforts in modification of the transporter in the wheat? So threshold level of nitrogen may be available to plant. What do you think?
Actually, in case of wheat or even all cereals, so it is very inefficient. First of all, we don't have the control like even plant, how much plant shall take nitrogen even for photosynthesis. We put lot of nitrogen in the for the photosynthesis machinery. We don't need that much. So then ideally plant shall remobilize that nitrogen to grain actually on time, because if it is not mobilizing to grain, we lose it in the straw, and that is again causing a lot of problems for us. So those transporters, there are several known by different workers around the world, but nothing efficient is happening in terms like mobilizing it and where we want it to go into the grain at the end. We don't want to stay it in the leaf or within those chloroplast machinery or photosynthetic machinery. That's why, sir, recently there is one good study where they have identified those genotypes which are less green, where they don't have more that chlorophyll. They are more efficient because they have shown a mechanism where they translocate more of this nitrogen into grain, so they stay less green. But these are very important genotypes.
Okay.
Maybe we need to do a lot of things, sir. This is the area which is picking up now in view of the nitrogen, importance of the nitrogen, again in view of the recent wars, etc. So it is again
Right.
a lot of things need to be done.
Another basic question you can say at my end, is there any other element which may induce or suppress nitrogen use efficiency? Have you reported?
There is a
Yeah.
correlation between zinc and iron, but nitrogen,
Yeah.
sir, you can please.
Certainly.
We have not, sir, seen these interactions. There may be, sir, several inhibitory effects which will, like several elements, if they are more, and then they may be reducing the nitrogen availability. But for us, like here, for a layman, if you don't have the water, that is the main constant here for us, because nitrogen remains in the soil, it don't go, plant is unable to uptake it from the roots, number one. If you have more rain in the beginning, it will leach out everything. So those are some of the major things which we know are affecting our programs. Like you can say from the distance, you have the nitrogen in the soil, but your plant is suffering because it's unable to take it because there's no sufficient water. So these are something, sir, which are visible, but there may be several like microorganisms. Like we know now, we have biological nitrogen inhibition is happening while several microorganisms, bacteria are involved in this inhibition of nitrification, etc. So if you have those phenomena happening in your root zone, then again your this nitrogen will be more available to plant.
Right.
So
But
there is a
these
good
things are happening
correlation
now.
between water and nitrogen. Do you mean to say, sir?
Yes, yes, yes. That's very important because
Mm.
we need to have good moisture in the soil.
Mm. Yes.
Okay, thank you. Thank
Okay,
you.
sir. Really, thank you for joining
Thank you.
with your busy schedule.
Thank you, sir. It was pleasure
Thank you.
interacting with all of
Really,
you.
thank
So
you.
hope
Dr.
to
Tarun,
see
could you
you
please-
again sometimes in future.
Yes. Yes, sir. Sure, sir. Yeah.
Thank you, sir.
So thank you, sir, for your presentation.
Thank you, sir.
Yes.
Thank you so much, sir. Yeah. Dr.
So-
Tarun, please introduce next speaker.
So now it's my pleasure to introduce our third speaker of the session, Dr. Dinesh Kumar, Principal Scientist at Indian Agriculture Research Institute. So Dr. Kumar is a distinguished agricultural scientist with extensive expertise in soil fertility management, rice agronomy, and organic agriculture. His work has significantly contributed to sustainable crop production, soil health improvement, and resource-efficient farming systems. He completed his higher education from G.V. Pant University of Agriculture and Technology and earned his doctoral degree from Indian Agricultural Research Institute, New Delhi. Throughout his career, he has focused on integrated nutrient management, sustainable rice production systems, organic farming technologies, and strategies to enhance soil fertility and nutrient use efficiency. As a scientist, as a mentor, and educator, he has guided numerous postgraduate and doctoral students and contributed extensively through research publications, innovations, and capacity building initiatives. His work aligns closely with the objectives of climate resilient agriculture by promoting environmentally sustainable practices that ensure long-term agricultural productivity and soil health. Sir, we are honored to have you today. Sir, I respectfully invite to share your valuable insights with the audience. Sir, can you unmute yourself?
Sorry. Thank you very much, Dr. Saab, and I'm thankful to the organizers also for giving me opportunity. Is it audible?
Yes, sir.
Okay. So can I play slides myself or?
Sir, if you can play, then it's good.
Yeah, I wish I wish to play. It's fine now. Okay.
Yes, sir, it's started.
Yes, sir. Yeah.
So friends, the topic of discussion is organic farming as a pathway to climate resilient agriculture. So why we are talking about climate resilient agriculture? That means there are certain problems with the existing agriculture or existing crop production. What are those problems with the existing agriculture is decline in factor productivity. Friends, we know that there are 3-4 important factors that are affecting the productivity and production: land, labor, capital, and management. So these are the four important factors or inputs or resources which are required by any agriculture. So in this case, the productivity of these factors is increasing. I can give you one example. For example, nitrogen is an input in crop production, which is purchased by using money. So it becomes kind of short-term capital, your seed, your fertilizer, your pesticides. So the productivity of these inputs is declining. Means with the passage of time, the productivity of nitrogen is declining. For example, 30 years before, 1 kg nitrogen would give you about 32 kg grain of rice. Now same 1 kg nitrogen is giving 15 to 16 kilogram grain of rice. That means productivity of this factor has decreased. It also suggests that if productivity has decreased, that means it is creating more problem to the environment. It may be polluting the environment, and simultaneously there is increased cost of production also. Now to get 32 kg grain of rice, you have to apply double quantity of nitrogen. That is 2 kg nitrogen will give you 32 kg grain. That means cost of production is increasing. So this is related to conventional agriculture, and soil health is also declining. There is a lot of data from Ministry of Agriculture itself, which suggests that larger parts of Indian soils are deficient in NPK, carbon, micronutrients. A lot of problems related to soil fertility or health have cropped up. I need not to talk about environmental pollution or kind of things which are happening to the environment. Reduced biodiversity is another issue. If you go to Punjab, if you go to UP, if you go to any part of the country, you will hardly find any diversification of the crops. Most of the areas are occupied by monoculture kind of thing. Rice wheat system, maize wheat system, cotton wheat system. These kind of systems are dominating. Means we have very little chance for diversification of the crop. Just before Green Revolution in Punjab, they were taking 20 different kind of crops. Now they have reduced only to 6 to 7 crops. That means crop diversity has declined. If the crop diversity has declined, that means your soil biodiversity will also decline. And this is not good for sustainability. This is not good for soil health. It is not good for crop production also, because the cost of cultivation will increase if we reduce the soil biodiversity, if we reduce the plant biodiversity. Now more and more incidences of insect pests and diseases are being experienced, particularly due to climate changes. Many people assign the increased incidence of insect pests and diseases and also weeds due to climate changes. Unsafe food, everybody would agree here that whatever foods are sold in the market, many of them are having more pesticide residues. You take any example, you will find in news, everyday news, you will find that so many samples were found containing pesticide beyond the permissible residue limit. So I need not to go into the details, but we understand very well how much pesticides are laced in our food. Reduce nutritional security or quality. If we talk of quality of Indian foods, there are many reports from National Institute of Nutrition, Hyderabad, which clearly suggest that during last 28 or 30 years, the food quality in India has declined. The protein content in many grains, including lentils, have declined. Iron content in apples, tomatoes have declined. Vitamin content in many fruits and vegetables are declined. These are very serious issues studied by National Institute of Nutrition. So therefore, the quality of the food is also reduced, and partly it is attributed to climate changes. So overall, what I suggest here, that the conventional agriculture is not only leading to problems in environmental pollution or decreased income of the farmer, but at the same time, the food becomes unsafe and also nutritionally poor food. Anybody interested can have data on current nutrient content in Indian foods from National Institute of Nutrition. So there are two causes for decline in food quality, or the nutritional composition of Indian foods is declining. For example, first reason is assigned is that our soils are not rich in nutrients. They are deficient in nutrients. Half of the Indian soils are deficient in zinc, and some are deficient in iron, manganese, copper, boron also. So this is one reason. If our soils are deficient, how come these soils can provide the plants or the food items which are rich in these nutrients. If my soil is rich, deficient in manganese, then my plant items will be deficient in manganese. My food will be deficient in manganese. And the people who are relying on that food will also have manganese deficiency. Supposedly, they will have manganese deficiency. So this is how the link is there, link of soil health to the human health. Then climate change is also diluting the concentration of nutrients in Indian foods, maybe elsewhere also, but you can see the carbon dioxide concentration in environment is leading to increased carbon dioxide synthesis or photosynthesis, which is causing more carbon fixation in the plant and reducing the concentration of the nutrients. For example, there are a number of studies. In many studies, it has been found under controlled conditions that the micronutrient content in our food declined due to rising level of carbon dioxide in the atmosphere. So what is overall happening? I try to summarize in this one small slide. So we see that why this environmental pollution is happening. This is the result of human activities and partly due to agricultural activities. So we have soil pollution, we have water pollution, we have air pollution, we have food pollution. Food pollution in the form of unsafe food. So just take the case of air pollution. Air is polluted. That means our greenhouse gases are there in much higher concentration today than they were before. So these presence of greenhouse gases in atmosphere is causing global warming. And this global warming is leading to climate changes. I think everybody knows this. I'm just making a story out of it. Affecting the climate changes, and then these climate change will affect back the human beings, the all kind of lives on the earth, and it will also affect the crops. So finally, these climate changes will result in decrease in yield and decrease in quality. So what can be done? Overall, what can be done if we expect that these climate changes will affect our yields, our productivity, and quality of the food? So definitely we need some climate resilient technologies that can help us to at least maintain the current level of food quality and try to increase little yields also. So overall, we see unsafe food, nutritionally food, or poor food. So what it causes, it will result in more diseases or more health issues in human beings. And we are visiting hospitals now. You go any hospital, you will find large crowds outside the hospitals. They are paying money, lot of money they are paying on medical bills. So this is really very serious threat, decrease in quality and more pesticide in foods. So what can be done? Alternative could be that we need to make our agriculture resilient to these changes, these kind of changes, so that there is little impact of these changes, and we can improve the food quality and quantity by following certain practices in agriculture. So any agriculture that helps our farmers or our farm production, our farmers continue stable crop production despite changing climate conditions. Means our agriculture, our production remains unaffected due to climate changes. Then that kind of agriculture will be climate resilient agriculture. So such agriculture can withstand, it can adapt and recover from the climate related stresses. Definitely stresses are going to be there. For example, high temperature stress, waterlogging stress, or water scarcity may be there, water stress may be there, floods may be there. A lot of problems are going to increase, but agriculture will not change. It will not be affected negatively. So what are those kind of agriculture, or what are those kind of practices? Many people have suggested long list of practices, but I have sorted out them, like integrated crop management could be one option. And then integrated farming system, organic agriculture could be one option. Conservation agriculture is also there, agroforestry system, and of course, precision agriculture. So these are some of the listed methods of agriculture, methods of farming that can partly or that can fully resist these climate changes, or can be helpful in resisting these climate changes, or these are climate resilient agricultural practices, I can say. But important point here is that one of these practices may not be applicable to all Indian conditions. So they cannot fit every soil, they cannot fit every climatic conditions, every farming situation, etc., etc. So there may be some specific niches or niche areas where these practices can be followed. For example, organic agriculture. It cannot be horizontally spread in whole India. There is no need, because under those conditions we may use different methods of farming or different methods of agriculture. So here I just want to restrict myself to organic agriculture. How organic agriculture can be a climate resilient practice. That is the issue. And I need not to explain to this group what is organic agriculture, but basically here we exclude the use of chemicals, synthetic pesticides, and also it is environmentally and socially responsible approach where human element is always there. Human element has to be there in agriculture. So in organic agriculture, we must take care of all the human elements, all the animal elements, all the plant animals and elements, and all the microbial elements along with the environment, so that we get the best combination, we get the balance out of it. And such agriculture also preserves the reproductive and regenerative capacity of the soil. Means our soils will continue to enhance their health. Similarly, there will be good and nutritious food for the people. So that is actually organic agriculture. So what is there in organic agriculture? So organic agriculture, when we practice under agricultural situation, under farming situation, that is called as agro-ecosystem. Means the farm where farmers are doing cultivation are called as agro-ecosystem. These agro-ecosystems are artificial system. So we need to apply certain principles of natural ecosystem into this artificial ecosystem so that it can be converted easily to the organic farming system. So we need to learn lessons from the natural forest system, for example. Natural forest system, they are very strong, they have very good buildup, and nutrient cycling happens in natural ecosystem. In natural ecosystem, the systems are closed. There are no losses of nutrients. There are no losses of biodiversity also. So they are self-sustaining system, natural system, because nutrient cycle happens naturally there. But we need to imitate it. We need to mimic it under the organic farming system. So here we need to recycle the nutrient. Taking a leaf or taking an idea from the natural forest ecosystem, we can recycle nutrients in the form of crop residues, in the form of compost, in the form of manure. Similarly, in the natural forest system, diversity is the rule. That is why these systems are very strong. Go to Western Ghat, go to Amazon forest. They survived centuries because of diversity. They have plant diversity, animal biodiversity, or you can say microbial biodiversity. Very rich biodiversity. So we want to apply these principles in the agro-ecosystem under organic system. So organic farming cannot survive without crop diversity. If we increase the diversity on the organic farm, the chance of success can be more. The yield gap can be reduced only by crop diversity under organic system. So crop diversity is necessary. Similarly, protection of soil. Under natural ecosystem, there is no soil erosion. Whatever erosion is there, that is called as geological erosion. But under agro-ecosystem, we got accelerated soil erosion. So under organic system, we need to preserve the soil, and that is soil protection. Soil protection can be done by adding organic matter, by using mulching, and by using cover crops. Similarly, here you need to maintain eco-balance. In the natural system, ecological balance is there. Every population, whether plant, animal, or microbes, they are at a balance. They are not in excess, they are not in shortage. So nature takes care of the things. If population of one tree is increasing, it will create some disease, some insect pest, so that population is controlled. So ecological balance is maintained under natural ecosystem. And we want to apply this knowledge to the organic ecosystem. So there we need to use these biocontrol agents. So certain biocontrol agents can be practiced, and we try to increase the population of friendly insects that can automatically control the harmful insects. So this is how we learn from nature and we apply to the agro-ecosystem. That is actually organic farming system. So therefore, the basic practices in organic farming should be encouraged, like recycling of nutrients, soil protection, crop diversity, and of course, the biocontrol. Now, definitely we need to justify whether the organic farming can be climate resilient. Does it able to provide sufficient productivity, sufficient yield? Does it provide nutritious food or safer food? Or does it provide sufficient income to the farmer? Or does it maintain or improve the soil ecosystem? Can it give us clean air and better climate? Can organic farming give us better groundwater and better surface water? If it can qualify all these points, then definitely it can be considered as a climate resilient practices. And friends, remember, all these climate resilient practices are also called as sustainable agricultural practices. They are basically sustainable agricultural practices. So we will just see one by one how the productivity is there, how much productivity is there under organic systems. So many papers, many reviews, many authors, many researchers have done a lot of work. I'm just summarizing here the results. One paper came long back in Nature, and that paper very clearly said that yields are comparable under many situations. However, under some situations, the yields are low in organic system. However, the extent of decrease in yield will vary with the soil type, with the climate, and with the management practices, and also with the cropping conditions. So you can see here, particularly yield or productivity of different crops during conversion periods decline. It is experienced by large people. Many scientists, many farmers clearly say that yield decline is there under conversion period. So they are lower definitely compared to conventional farming. And they said this yield is very subjective. Yield decline is very subjective. Under rain-fed conditions where legumes are grown or oil seeds are grown, the yield decline may be only 5% because those kind of conditions are quite close to natural or quite close to organic farming system because they are using very less chemical, very less pesticide. Therefore, yield decline is lower under those conditions. Similarly, there may be higher yield decline, maybe 34%. 34% decline may be there even after the conversion period. Even after the 2 to 3 years period of conversion, the yield decline may be 34%. This is particularly true with the intensive farming system or irrigated farming system, where farmers use more pesticide, where farmers use more chemicals. What happened? These soils have become addicted to the chemicals or pesticides, and they do not respond to organic inputs very quickly. So it takes time for them to get recovery. It takes time to maintain or improve the biology of the soil. So yield reduction are more under intensive agricultural systems. Under certain conditions, if we can manage good crop management and growing conditions are improved, and with the passage of time, organic farming can result at least equal yield to the conventional farming systems. So yield it stabilizes after certain number of years, say 4 years, 5 years, yield will stabilize and it will become equal to the conventional farming. This is experience of large number of people. So over time, organic farming improves soil organic carbon, soil aggregation. These are the benefits to the soil. And if soil is benefited, the plants growing there will also get benefited. And the human beings, those who are consuming these products will also get benefited. And of course, the environment will also get benefited. So these changes actually improve the crop yield. Now, what are the conditions or what are the crops which get more benefit? It is not that organic farming will give you better results under every kind of crops. For example, crops like sugarcane, potato, rice, high-yielding rice, wheat, those kind of crops require large nutrients and they are affected by major insect pests and diseases. So therefore, they perform not that much better as other crops. Like in rice, Basmati rice has performed better because of low nutrient requirement. Soybean is an ideal crop. Soybean, groundnut, pulses are better. Vegetables, certain vegetables like tomato, cole crops are good candidate for organic farming. And in my view, spices and medicinal plant are the best bet for organic farming. So because spices are in low volume and high value crops, so high value crops with low volume are more suitable because these crops require less nutrients, and they also require less insect pest management practices. Millets are also important, and many farmers are turning up to the millets and looking up to the millets. And now we have very important emphasis on the millets also by the government. And now farmers are thinking of introducing millets under organic systems. And friends, this one slide completes everything about organic farming, how it is resilient. So you can see this slide, it is from Rodale Institute of USA. They've done long-term studies, and you can see here that yields are comparable. This green bars about organic farming and gray bars about conventional farming. So you can say yields are comparable, same yields. And if you see the profit is more in green organic farming, energy input is less, and the pollution is also less in organic farming. So therefore, it is definitely a kind of climate resilient agriculture, which is emitting less gases, using less energy, giving more profit, and giving equal yield to the conventional farming. This is our trial. We have been doing a long-term trial since 2003 at IARA. Now, this trial has completed about 23 years. Anybody has any interest to see this trial, you can come. Now we are having mung bean and sesbania in the field. And then during Kharif season, we will have baby corn in the field. So anybody interested can visit IARA and can see these trials. We have two long-term organic farming experiment here. So our experiments suggested that we can get good yields under organic farming for basmati rice as well as for wheat. And also diversification through legumes is more beneficial in organic farming systems. And similarly, large number of trials have been conducted at Modipuram. Indian Institute of Farming System Research has a long-term project, network project on organic farming. That project is called as Network Project on Organic Farming, funded by ICR. And that project has 20 centers, pan-India 20 centers, and they are doing research since 2004. And they have come up with package of practices for organic farming. A lot of work has been done, and they have concluded that after 8th year, you can get the similar yield in organic farming as conventional farming after completing the 8 cropping cycles. And of course, in subtropical India, in winter crops like wheat and some other crops, the yield is still less under organic farming, say 8 to 10% less yield. So you can have this presentation and go through all these slides. So why organic farming increases yield? Why it becomes slightly higher than the conventional farming? So mainly because of higher soil organic carbon content, better microbial activity, and legume-based rotations. So it is really compulsory to include legume crops in the rotation if we want successful farming or successful organic farming. We need to use compost and farmyard manure, green manuring, mulching. The result on crop diversification is the main key for the success of organic farming. We must use biofertilizer and biological pest management practices. So therefore, soil health improves, environmental health improves. And there are a number of reports which say that organic farming also results in better food quality and our foods are safe. And antioxidant contents are more. Most studies across the world, including in India, suggested that organic food contain on an average 50% more antioxidants, which have several health benefits for human beings. And income, many data from India and from our experiments suggested that cost of cultivation is slightly lower than conventional farming. Yield gap may be there, particularly in the transition period. And of course, we may get 20% higher prices for organic products, and income increase may be 10 to 20%, very subjective. And we are exporting, though very small money we are getting by export of organic products. And also, there are certain specific conditions where farmers will get more profit. Friends, it is very subjective. Government policies also affect the profit of the farmer. I give you one example. For example, Punjab farmers are getting free electricity. So their cost of cultivation on water management will be less. In another state, farmers are not getting subsidy on electricity. Then they will be using diesel to irrigate their crop. So then the cost of cultivation of those farmers will be more, those who are using diesel to apply water, to apply irrigation. So it is very interesting. So condition-wise also, the economic varies. Rain-fed and low-input areas, they will benefit more. Tribal and hilly regions, they will benefit more from organic farming. Spices, tea, coffee, fruits, vegetables, medicinal plant will give you higher income. Millets, pulses, basmati rice, they will give you higher income. If you go for integrated farming system, it will give you, it is very important approach for organic farming. And of course, if you can make FPO, then also your benefit or profit will increase, because then you will have negotiating powers. If you have cold storage facility, you can have more economic benefits. So number of factors affect the economic benefits. And of course, soil ecosystem is also improved. Carbon content will improve because you are adding organic matter. Water use efficiency also improves because water storage capacity also improves. You can reduce the number of irrigations if you follow organic farming, because the water holding capacity of soil increases. Soil erosion will decline, and definitely organic farming is good for carbon sequestration. And there are number of reports, number of papers, our report also, which says that organic carbon increases, and it is natural also, because we are adding organic matter by farmyard manure, by green manure, by compost, etc. So organic matter content is bound to increase, and we will get better air. And similarly, the water quality of the surface water, groundwater will also improve. The nitrate leaching will reduce, and greenhouse gas emissions will also reduce. So therefore, the quality of the water will improve. And you will have several questions on this. I have put forward certain questions to you. If you know the answer of these questions, you need not to ask, or you can ask any question out of these, or you can ask questions of your choice. You are free. For example, why fertilizers are not allowed in organic farming? Why GM crops are not allowed? Is organic farming sustainable? And how about quality? Plant does not discriminate between nitrogen coming from fertilizer or organic sources. Is organic farming profitable? How insect pest, disease, weeds can be controlled? Can organic farming mitigate climate changes? So these are the questions from my side that audience would like to ask. In addition to this, you can ask any question of your choice. You will be most welcome to ask it. Ask your question, and now I can unshare it. So stop share. So your questions are welcome.
Thank you, sir, for the excellent presentation. We shall now open the floor for interaction and questions.
Tarun, read one question in chat box. One has
Yes, sir.
dropped.
Yes, sir.
So Dr. Jimmy John is asking.
You can read it. I don't know much about this chat GPT. This you ask me a straightway question. The anchor. Yeah.
Yes, sir. I will read it out for you.
Yeah.
Yes, sir. So it's they're asking. It's often seen that plants grow well in diverse, in that provide very little substratum, while plants in nutrient-rich pot culture often have stunted growth. Comparatively, this is also seen in field cultures. Please highlight what we are doing wrong.
I request you with folded hand to repeat the question, please. It is very
Actually, some plants are growing well in the substratum, in the debris.
In the debris, they grow well.
Debris.
Yeah.
In the debris.
Normal debris. Yeah.
Let me read the question in the revival manner. Sir, Dinesh sir, it is a question. It's often seen that plants grow well in debris in that provide very little substratum, while plants in nutrient-rich pot culture often fail and have shown stunted growth. So please have insight on this, where we are going wrong. The question comes to us.
Yeah, it is good. Debris means they are growing in field conditions and they have used some residues. And plants are happy under those conditions, but they are not performing same as under pot conditions, where we can provide some nutrient-rich medium also. So definitely see the conditions of soil profile. If we go to the soil, the natural conditions are there in the soil. And nature has given us to grow the plant under natural conditions in soil. So this soil profile also contributes to better root growth. Plant may get better oxygen and better sunlight. And there, if you grow in pots, that means you are growing under greenhouse conditions. It can be also understood. So in organic farming, this greenhouse conditions are also allowed in organic farming. So they have made some compromises. So you are using some artificial medium there. It can never be compared with the natural things. So in nature, of course, we know that plant require 17 essential elements, but we are not sure. The list is endless. In America now, they have increased, they have added cobalt also as essential plant nutrients. I don't know whether cobalt has been added in pork culture or not. And if you see, they are 20 now. Some American Chemist Society, they increase the nutrient number. So in soil, plants are not getting just these, because microbiology of pot may be different from the field. I tell you, microbiology also plays important role, and the kind of taste of food that is grown in natural condition will never come in pot conditions, because their microbes may release variety of amino acids, variety of hormones, and they will make balanced growth. So this is in totality, we are taking totality. That is partly artificial kind of things, and we cannot get the same as we get in natural systems.
Right.
For detail, they can send me email. I will send the detailed answer.
Right. Second question, sir, from Sonia. Thank you, Professor Dinesh. Can there be something 100% organic?
See, it is just assume that it is organic. First thing is that you rely on certification. Certification is necessary. And certification is done only for the process, not for the product. My product may still contain pesticide, and although it is certified, there is no guarantee that organic products will not have pesticides. They may have, and sometimes some mischievous people may be there, so it happens. But I tell you, there are many natural ways through which pesticide can enter our organic food. Even if I do it myself, even then I will get some pesticide residues in it, because I do not have control on the acid rain. I do not have control on the rainfall. When rainfall comes, rainfall may contain some poisonous material. You know about air, how the rainfall comes. It mixes with air, then it comes to the soil. So some pesticide may come from that. Some pesticide may come from canal water. Sometimes farmer use groundwater. If my groundwater is having pesticide and I am doing organic farming, I cannot get distilled water for irrigation. So they may come from there also, and they may come from seed also. Sometimes in organic farming rules, the first time you can use the chemically seed from the conventional farming, provided it is not treated with the pesticide. So that conditions are also there. So therefore, the pesticide have a chance to enter the food chain. And also during the marketing period, storage period, some farmer may use pesticide, or some retail people may add pesticide. Thank you.
Tarun?
Thank you, sir. So I have a question actually.
Sure.
So actually, as we know, the most of the varieties in today's days, we are using high yielding varieties that is suitable for the fast release of nutrients from the fertilizers. But when we apply the bulky organic nutrient sources that are releasing nutrients at a slower rate, sir, how can we fulfill the nutrient requirement of those varieties or crops?
Yeah,
Yes,
really very relevant things you have asked. So first thing is, if we see from organic perspective, that
Yes.
we cannot avoid organic manures, because they are the major source of supplying nutrients. So we need to actually diversify the nutrient sources. First point is that we should not solely rely on farmyard manure or compost. We need to use vermicompost. We need to use crop residues directly into the feed. We need to use some biofertilizer. We need to use oil cakes.
Yes,
For
sir.
example, neem oil cake, non-edible oil cakes can be used. And also there are certain minerals which can be allowed. For example, gypsum is allowed in raw form. Gypsum can be used. It is source of sulfur, and it can help in decomposing the organic matter faster if we add sulfur, because it will acidify the environment, and decomposition process can be accelerated. We can use rock phosphate in raw form is allowed.
Yes.
And there are number of minerals, even potassium chloride, if it is used in natural form, which is dug in mines in Germany, in China, in Canada. They have minerals which are rich in micronutrients and so on. So those minerals can be imported. Even in India, there are some sources, some minerals. We may think of using them. So by taking all possible sources, we can sort out this problem. Secondly, we should take the crops and varieties which require less of the nutrients. I already told that we should not go for sugarcane, potato, wheat, paddy, high-yielding varieties. We can start with the spices. We can start with mooli, radish does not require much of the. So those kind of vegetables, spices can be preferred. And within these, we can take the varieties which are of shorter duration. Take the case of rice varieties. Rice variety mature in 145 days or 150 days, basmati varieties. But we have variety which matures 110 days.
Yes.
Their nutrient requirement is less. Similarly, in other parts of the country, we have our largest diversity in case of rice crop. So there are certain genotypes which mature in 90 days, 100 days. We can go for that. Red rice, black rice, so many things are there in our country. We can take benefit of that. Thank you.
Thank you, sir.
From chat box, there is another question.
Yes, sir.
Compared to synthetic fertilizer, organic fertilizer required in bulk amount to supply the same amount of nutrient.
Yes.
How can we manage the bulk amount of organic fertilizers in spite of reducing animals like that?
Yeah, this is the problem. This is the biggest issue with all of us, that we have mechanized our agriculture, particularly in Punjab, Haryana, or some other parts of India, and only milch animals or milch cattle are kept by farmers. So definitely we need to look into this. So we cannot supply farmyard manure overnight. But farmer need to be very active. I tell you, there are certain goshalas, and for them, disposal of cow dung or disposal of dung is an issue. So let us contact those farmers. My brother is doing conventional farming, and he had made contact 30 kilometers away from our village with one goshala. And there they are giving very cheap dung. So that could be one of the source to contact the dairies, to contact the goshalas. We can buy it. And secondly, we need to grow green manure crops. We need to use biofertilizers also. Definitely, we need to go for integration of organic sources. We need to go for green leaf manuring in some cases wherever some vegetation is available. Biofertilizer are good source. And how long we will continue with the fertilizer? Now you see government is also coming in a big way to reduce the fertilizer. So we cannot rely long on the fertilizer. Someday we need to reduce the use of fertilizer. Government has set the target to reduce fertilizer need by about 25% by 2030, if I am correct. So less fertilizer will be available in the market. Then whatever is available, we have to work with that. So better to get ready in time, to wake up. It is a wake-up call. So we need to think on those lines, sir.
The farmers are still, some of them, they are not having awareness. Still after harvesting, they are burning the field.
Yes.
And they're destroying the normal flora and fauna. So can you suggest something about that? Because some farmers are still not having awareness about the crop residues after harvesting, etc. Could you please suggest anything, sir?
Definitely, number of practical uses are there. And now there is significant reduction in burning of residues also. There's no doubt about it. And government has come in a big way. Now they are doing fine to the farmers. A lot of supervisions are there, and there are changes in the things. So first thing is that problem is that those farmers do not have animal. My village is in Moradabad district, and my father, my brother, they are feeding rice paddy to the animals. You cut it into pieces like any other fodder and mix with the green fodder in 50-50, in 25-75, and animals are happy. They can eat it. They can eat this. So it can be used as a good animal feed. They use bedding material for animals, and then it absorbs urine and cow dung get mixed, and they can compost it fast. Composting is faster. And another use is that cut it or mix it in the soil. Now, a lot of options are coming up with this happy seeder, and you can do sowing in residues now. In Punjab, this time I talked to several farmers. Now they are switching over to direct seeded rice. So in DSR, you have to directly do sowing of DSR can be done in residues.
DSR, yeah.
But not many farmers are burning wheat residues. Problem is with rice residues.
Yes, yes, you are correct.
Okay, Tarun, you may lead, please.
Yeah.
Thank you very much, sir, for your insightful and enriching presentation. You're valuable.
Just Tarun, I wish
Yes.
to say one line to thank to your team, to Dr.
Thank
Karthi, Dr. Chaudhary, and Dr. Deepshika, yourself, everybody who has invited me, given me an opportunity. Thank you to all. And they can send me questions through email. I will respond all questions. Thank you very much.
Thank you so much, sir.
One more line I would like to say, Dr. Dinesh. I am from Pantnagar also.
Oh.
BN Johari student.
Which year, sir?
I think I may be the junior of you. I did PhD in 2000 around 2003, 2004.
Okay, I did MSc in 1992.
Yeah.
And then I did PhD from IRA, sir.
Great.
Anyways, we are alumnus.
College of Agriculture.
Yes, I was.
Okay.
Yeah.
So
Agronomy mein tha, sir.
Yeah. We shall meet soon. We shall meet soon.
Sure, sir. Sure, sir. I will be happy.
Thank you.
Thank you so much, sir, for your sustainable agriculture related presentation. Very nice. Thank you.
Okay, thank you, sir. So, as we conclude session 1 of day 4, I sincerely thank all our esteemed speakers for sharing their valuable knowledge, experiences on recent advancements in genomics, genome engineering, molecular breeding, and sustainable agriculture. I also thank all participants for their active involvement and enthusiastic participation throughout the session. Their discussions have highlighted the immense potential of modern genomic technologies in developing climate smart and resilient agriculture systems capable of addressing future food security challenges. Thank you all for your presence and engagement. We look forward to your participation in the upcoming sessions. Have a wonderful day ahead. Now it's time for the lunch break. I request you all to join at 2 p.m. Thank you very much.
Today's second session. On behalf of organizing committee, I extend an heartfelt welcome to our notable speakers and participants. Thank you for joining us. I hope you find today's session informative, engaging, and inspiring. It is a pleasure to welcome all the distinguished resource persons, faculty members, scientists, researchers, scholars, and participants to the 4th day's faculty development program. Following an engaging and enriching first session, we are delighted to continue this academic journey with the second session of FDP under the theme 4, focusing on recent advancements in genetic and genome engineering for climate smart agriculture. This program serves as a platform for knowledge exchange, interdisciplinary learning, and meaningful discussion on informative approaches to addressing the contemporary agricultural challenges. We look forward to another day of insightful deliberations and fruitful interactions. To commence this session, it is my immense honor and privilege to introduce our first prominent speaker for this session, Dr. Saloni Mathur, scientist by National Institute of Plant Genome Research. Dr. Mathur is an accomplished plant molecular biologist and scientist working in an area of plant stress biology and climate resilient crop research. She is currently serving as a staff scientist 5 at the National Institute of Plant Genome Research, and her academic and research career has been strongly focused on understanding molecular mechanisms governing a plant responses to environmental stress, particularly heat stress under changing climatic conditions. Dr. Mathur completed her graduation in botany from University of Delhi in the year 1997 and subsequently obtained her master's in the year 1999 and PhD in 2005 in plant molecular biology from the Department of the Plant Molecular Biology, from University of Delhi South Campus. She later served as a research scientist at the same institutions and at the Interdisciplinary Center for the Plant Genome before joining NIPGR as a Scientist 3 in 2013. Her research preliminary focused on understanding how plants respond and adapt to the heat stress, which has become a major challenge for a global agriculture due to a climate change. Dr. Mathur Group is particularly interested in a role of microRNAs in relating to heat shock responsiveness genes in a tomato and other crops. Through comparative molecular analysis and contrasting tomato cultivars, her team investigates the microRNA-mediated regulatory networks involved in a flower and fruit development under the elevated temperature. The long-term objective of her research is to identify the key genetic regulators that can be manipulated to improve the heat tolerance, crop survival, fruit quality, and yield stability under the adverse climatic conditions. Her work integrates plant genomics, molecular biology, and stress signaling research to develop the innovative strategies for climate resilient agriculture and sustainable crop improvement. With these words, I once again extend my heartfelt welcome to you, ma'am, and invite you to address the gathering. Over to you, ma'am.
Thank you so much. Can you hear me, first of all?
Yes, you are audible.
Okay, I'll share my slides. Can you see my slides?
Yes,
Yes,
madam.
Yeah, okay. So shall I start? Or we are
Sure,
little
sure.
ahead of
Please,
time?
please go ahead.
Okay. So at the outset, I would really like to thank the organizers at Amity to have thought of me and invited me so that, you know, I can share some of the research work that is going on in my laboratory. And in doing so, I would be covering many slides. So what I have tried to do is that I am going to give some very basic things, which for you all would probably be just a revision. But I think revision always helps. So we'll start with some basic things, and then I will end with the kind of work that is going on in my laboratory. So basically, yeah. So we all know that the initial central dogma of molecular biology was unidirectional, wherein DNA, while it was able to replicate itself, but in one direction, it was giving out the RNA molecules, which in turn were being translated and proteins were being formed. Later on, with the discovery of certain RNA viruses and its subsequent mechanism of action, this central dogma got changed, wherein now this directionality was challenged. And while the viral RNA itself was able to replicate an RNA to RNA using RNA-dependent RNA polymerases, there was another action using the reverse transcriptase, wherein RNA could also be converted into DNA. So basically, the hallmark of all these experiments in molecular biology in the 2 to 3 decades back was concentrated around the prediction of proteins and finding their functions and finding their mechanisms. Till the time a milestone and fundamental event happened where the human genome was sequenced. So when the human genome was sequenced, which is roughly 3 billion bases, it was presumed that being such complex organisms, there will be many proteins in the human organism. But it was a surprise that only around 2% of the genome that was assembled actually coded for something which could be translated into proteins. So what does that mean? Either that the 98% of the genome that is present is just junk, doesn't do anything, but a more likely scenario at that point was to relook into the genome to find out whether there is some other kind of a genetic information that is hidden and it is not really the junk DNA. So to answer this, many international laboratories across different countries joined hands, and a project called as ENCODE was started, where the international, which is the International Encyclopedia for DNA Elements, to decipher that what is the human genome encoding. And they were able to actually ascribe some kind of a biochemical activity to 80% of the human genome. And this was done by finding any kind of a reference to a transcriptional activity, a presence of a transcription factor on the chromatin, the structure, the histone modifications. So yes, the clue started to come that, okay, it's not only the junk, this may be doing something. And this kind of scenario is not restricted to humans, with so many genomes having been sequenced subsequently and using exceptional computational analysis that is available. Several plant species were also in the same category, wherein a large part of the genome is not apparently coding for a protein, but it may be associated with some kind of a transcriptional activity. So basically, what is this junk DNA? It basically, the so-called junk DNA is actually composed of pseudogenes, transposons, repeat elements, several regulatory elements, which include the cis elements, the enhancers, the promoters, and other regions where protein associate, as well as the non-coding RNAs, which brings us to the topic basically of today. And what are these non-coding RNA? They are actually the functional moieties, and they also, get diverted into two aspects: the housekeeping ones and the regulatory ones. So the housekeeping non-coding RNAs, we all know, are the ribosomal RNAs, the tRNAs, and the SNO RNAs. But today I'm going to focus on the regulatory non-coding RNAs, which are also many kinds. But I have written about the microRNA, long non-coding RNAs, and the siRNAs. So what are these non-coding RNAs? So the small RNAs, which comprise of the microRNAs and siRNAs, they are actually a pool of silencing agents, which are around 21 to 24 nucleotide long RNA molecules. So if you look at, let's say this is the distribution of reads from a small RNA transcriptome, and one would find that in the plant systems, the predominance of the number of molecules vis-a-vis the length is between the 21 to 24, with the 24 nucleotide being the maximum. And the other, besides the small RNA, are the long non-coding RNAs, or they are the LNCs. And they are arbitrarily sort of defined as some transcripts which are more than 200 nucleotide long, and which really do not have any discernible coding potential. So a cutoff for this is that, it should not be coding for any ORF, which is more than 100 amino acids. So the mechanism of action of these small RNAs is basically gene silencing, and it is through three modes. One is that it finds its cognate complementary mRNA and it cleaves it. Second is that with a more not a high degree of complementarity, it still finds a target and is having a role in translational repression. And the last one is that it is actually doing a transcriptional gene silencing, wherein it can actually associate with certain proteins and then methylate the chromatin, and thereby bringing in silencing. So just a quick recap as to, what is the difference between these microRNAs and siRNAs and what is their genesis. So the basic primary classification of a microRNA is to an siRNA lies here, that this is a hairpin single-stranded RNA that folds back upon itself. And then there are others where the double-strand RNA is created owing to two separate RNA entities coming together. So the hairpin RNAs are the one which give rise to microRNAs, and its precise processing leads into so it has this is a precursor, it has a stem, it has a loop, which gives rise to these red ones, the mature microRNA and a star form. So this is a snapshot from one of the microRNAs we had been studying some time back, and to just give you a glimpse as to what this picture actually looks like on an NGS platform. So you see this black hairpin, that is the sequence over here, and these red ones over here are actually the microRNAs mapping over here, and the blue ones over here are these star molecules. So basically, the microRNAs are 1 minute. I think I pressed something. I need to go back. Can you see my slides?
Yes, madam.
Okay, but I think I pressed something else. 1 minute. How do I go to slide control? Actually,
No
Yes.
problem, madam. You may skip this slide.
No, I can see it now. Actually, a large part of my screen is getting blocked by the,
Okay.
yeah, now it's okay. I pressed the wrong button. Now I can see my slides. Earlier, half of my slides were visible to me. Yeah,
Okay.
fine. Sorry for the interruption. So what I was trying to tell is that the hairpin RNA, they are the mother of microRNA and the small RNA. And most of them are lineage specific. That means they have evolved in such a way that they are only present in some closely related species. Whereas there are some other imprecisely processed precursor hairpins also, but these so many small RNA mapping randomly on a precursor, they do not qualify as microRNAs. So the lineage-specific microRNAs, as you can see, here is a dendrogram of various microRNAs that have been discovered. And you can see that while most of them are conserved vis-a-vis the various phyla of the plant kingdom, indeed, there are some which are only, let's say over here, monocot specific, only which are dicot specific, and some which are more prominently present in the lower clades. And the other class of microRNA is the longer microRNAs that are being formed, which are not the classical lineage specific ones. And there has been experiments to show that these long miRNA, as they are called, have a role in depositing repressive chromatin marks on the heterochromatin and thereby bringing about silencing. So this biogenesis pathway of microRNAs is pretty complicated with many proteins involved in it. But the main thing to remember is that the double-stranded hairpin loop is being cleaved by a protein called as dicer in animals and dicer like DCL in plants. And then there is a risk complex, which is the RNA-induced silencing complex, which actually is taking the mature microRNA and target into its complex for the cleavage. And a very simple cartoon over here to basically show that there are slight differences between the microRNA biogenesis and mode of action between the plants and animals, wherein in plants, DCL is doing two jobs. It is cleaving the primary into a precursor microRNA, as well as cleaving this head to make the double-stranded structures, which in turn are transported to cytoplasm. And the major mode of microRNA action in plants is mRNA cleavage, which has a high complementarity level. On the other hand, in animals, besides the dicer, it is the job of drosha to do the primary to precursor conversion. The precursor itself travels to cytoplasm, and the dicer then does its subsequent job, wherein in most of the cases in animals, the translational repression is the major mode of action of microRNAs, showing only partial complementarity to the mRNA. Now, the protein AGO actually got its name from plants, wherein a particular mutant of this particular gene, which is called as AGO1, named after the octopus Argonauta argo. It resembles because of the kind of leafy radial leaves that it has. Now, AGO1 is the key protein that is of the RISC complex, and that preferentially binds to different microRNAs, where different AGO classes in plants, like 1 has preference for 5' uracil, AGO2 favors siRNA 5'A, so on and so forth. And as far as the mechanism of microRNA and AGOs goes, then I have talked about a simple cleavage, where in majority of the case, AGO1 plays a role. Then there is a translational repression. Again, AGO1 plays a role. Then how else does an AGO, affect a microRNA regulation? In case of Arabidopsis, microRNA 165 is chelated by AGO10. It decoys it away, thereby reducing the actual levels of 165 that can be incorporated into AGO to further downstream regulate its target gene. And in rice, AGO18 is able to, sponge up microRNA 168. It takes it away. This way, microRNA 168, which I must tell you, actually targets the AGO itself. So the target of microRNA 168 is AGO. So it is able to decoy this in case of viral defense systems. Then there is an AGO4, which has a role in the long microRNA mediated DNA methylation, as well as AGO2 has a role in double-stranded DNA repair. So coming to the other class of the small RNAs that I was talking of, the siRNAs, and just a quick recap, let me tell you, these are not from a single molecule that falls back. They are two separate molecules that come together to form a double-stranded structure. And they themselves can be of various kinds. So one is that heterochromatic siRNAs. So as you can see, these are siRNAs produced chiefly from intergenic regions or repeat regions. And then there are the, let's go to the below one first, the NAT siRNAs. And they are of two classes. The cis, that means they are just overlapping and they are forming natural antisense transcripts. Or they could be trans also, that these are repeat regions having this complementarity, but they are far away. The last class over here is the secondary siRNAs, wherein phased siRNAs are generated on double-stranded RNA, where microRNA is involved to generate this, which in turn regulates a trans-acting second target. So to make it little more clear, Here is a cartoon of these phased and tRNAs or transacting siRNAs. So it always starts with a microRNA. So you presume that there is a precursor that gives rise to a microRNA and it has a target, where the microRNA has a distinct site from a site where a siRNA, tRNA can be placed. So when microRNA sits here, it cleaves this target. Once this is cleaved, then there are certain RNA-dependent RNA polymerases which make this transcript double strand. Once it is a double strand, then it is targeted by DCL4. DCL then cleaves it into these small siRNAs of 21 nucleotides. It gets associated with AGO in a RISC complex and now cleaves a second target. So that is why it is called as transacting siRNA, because it involves a microRNA and then there are two different targets involved. So let us take some examples that how, if you had to identify a microRNA target pair, how would you go about it? So basically, let's say that if it has to be in heat stress, then you stress your samples. And what you can do is you can generate deep sequencing data of three kinds: the small RNA, the RNA, mRNA themselves, as well as something called as a degradome, which I'll just show in the next slide, which are the cut products of the microRNA target association. So basically, if you have to make a degradome library, you just have to understand over here that if a microRNA cleaves a target, then as opposed to a 7-methyl-guanosine that is safe over here, a 5' phosphate end is created. And utilizing this kind of feature, what happens is that we add an adapter, we make double-stranded libraries, we cut it, and then we sequence it. And using an entire pipeline of bioinformatics, wherein you place the original target, the cut products, and the microRNA one after the other to decipher that which microRNA could probably be targeting a particular step. So I would go a little quick on this, that, through digital profiles, one can pick a microRNA and target by selecting the anti-correlation in expression, and then further validate it by doing a 5' RLM which is a gene-specific form of the degradome that I just talked of. And this is how your, actual sequencing would look like, that if this thing was cleaved, there is an adapter and it joins like this. Once you will sequence these products, you will find a cut end like this, and this entire thing will be your microRNA sequence. So microRNAs, I don't want to go into detail into this, but they have fundamentally important role in all aspects of plant development, as well as in all kind of stresses and nutrition. And they are really important, wherein, you can just see this example of microRNA 164, that if this is the wild type plant, if you somehow mutate this microRNA, then you can see this jagged look in the serrated look in the margins. And one can say that how it is happening is through its target, wherein if you make a construct where the microRNA binding site becomes, it's in a way we call it as a resistant target, that microRNA can bind but not cleave, but gives the similar phenotype. This is the way to justify that a particular phenotype that we get is coming from either a microRNA or a target. And then microRNAs are mobile, and they have been their importance have been shown in leaf polarity. And microRNAs have been shown to play a role in vegetative to reproductive development from the juvenile to adult stage in flowering. I'm not going into the details of all this. And how microRNA processing itself could be regulated could be because of the differential presence of the kind of ago molecules that are present in a particular tissue, whether flower or vegetative, or the presence or chelation of certain biogenesis molecules that may hamper its biogenesis in certain tissues. Moreover, as I told you, AGO itself can be regulated by microRNA 168, and even DCL has a microRNA 162, and an interplay between these two can also regulate the actual expression of a particular microRNA and subsequently its target. So even though microRNAs have been called as non-coding, but recent papers have increasingly been showing that the precursor, upstream of the precursor, there are very short ORFs, and they themselves could be coding. And this has been shown in lateral root development. It has also been shown in other aspects of plant development and phenylpropanoid pathway. And coming to the long non-coding RNAs, as I said, that they are 200 base pairs long, and they are also classified as natural antisense. They could be intergenic, that means present between two genes. They could be intronic, or they could be associated with certain enhancers or overlapping with certain genes and thereby regulating its transcription. So long non-coding RNAs basically have many ways of, mode of action. You can say that it can either, form a double-stranded duplex from where if it is acting as an antisense RNA. It could be acting as a microRNA target mimic. It could be, inhibiting a particular protein-protein interaction. It could be bringing out a change in the structure of protein, or it could be, regulating an entire scaffold of protein complexes, or it could also be involved in protein relocation. So these are the examples of some of the long non-coding RNAs which have been instrumental in germination, root growth, phytohormone, stress response, and photomorphogenesis as well as leaf development. And here is an example of the first example rather of IPS1. Basically, this is a phosphate starvation response long non-coding RNA, which is actually working to regulate the toxicity of the plant in response to phosphate by regulating a microRNA 399 over here for the plant growth and development. So basically, my laboratory works on heat stress, and this is an important stress, and I don't need to really emphasize to this audience its significance. And we work on several aspects of HSFs. And we are working on HSFs, we are working on long non-coding RNAs and microRNAs. And so I'll just go very fast with some of the data that we have. So we generated some small RNA in response to heat stress and found several differentially regulated microRNAs. Then we cross-checked these microRNAs using RT-PCR, and we found the preponderance of this particular microRNA called as microRNA 169. So to our surprise, what we found was that tomato genome, let me tell you, is nearly, for 5 times of that of Arabidopsis. But in the database, there were 14 loci that were coding for this microRNA in Arabidopsis, but in tomato, there were only 5. So we took a bioinformatic pipeline and we identified many microRNAs by first confirming that they have the proper stem loop hair structures. And what we were able to show was that indeed tomato has more microRNA 169 members of the existing ones, as well as there were novel ones, and there were many more mature forms, and then there were many more loci. And what you can see from this phylogenetic tree is that even though, look at the green ones, for example, while most of them are grouping together, showing maybe some similar kind of a function, some of them have diverged using the precursor structure to gain some new functions. And these microRNAs, some of them were actually duplicated, showing segmental as well as tandem duplication. And rather, some of them were even, being transcribed as bicistronic units. So that is that, the same promoter was coding for 2 microRNA simultaneously. So basically, why does a plant have to have these duplications? What we were able to show over here is that it's through its promoter activity that you can see that there is a functional divergence, and the different promoter have been acquired during course of evolution so as to have different functions. So this particular microRNA family was highly expressed in the heat stress. So, out of the 16 ones that we had, 15 of them were highly expressing in the heat stress. We focused on that. And we saw that all the promoters have the cis elements, the heat stress elements, that are the sites for the heat stress transcription factors to regulate transcription. And by cloning all the HSFs that are present in tomato and all the promoters of all these 15 microRNA 169, we were able to show that some of these HSFs are able to actually bind to some of these microRNA 169s using various methods like the East-1 hybrid. Then there were the effector reporter assays in Nicotiana, and we also showed it by ChIP-qPCR. So basically, how does a system work? That there is a microRNA, it has an HSC to which HSF binds. If it is heat responsive, the precursor will be upregulated and the target would be downregulated. And indeed, this is what we found for all the molecules that I just showed in the previous slide. So if the microRNA is up and therefore its target would be down, essentially, what does it mean? That a reduction in the target should give us tolerance. So the 2 targets of 169, when they were downregulated, you can see as compared to the wild type plants, they are behaving much better than the wild type plant, suggesting that the targets of 169, the NFYs, are negative regulators of heat stress. And since we started the story with Arabidopsis, we also looked at it in the microRNA 169s in Arabidopsis, and we found that yes, all the 4 mature forms are basically heat upregulated, and all the precursors also have the HSE cis elements in their promoters. And again, we found certain interactions between Arabidopsis HSFs and Arabidopsis 169s. And we focused on HSF-A2, which you can say is sort of the master regulator of heat stress response. And we showed that, yes, this 169D, which is regulated by HSF-A2, is actually heat responsive. And if its promoter is put in the background of a mutant, then this expression is lost, suggesting a tight correlation between HSF-A2 and 169. Then, using different methods, we narrowed down on what could be the transcription factors that are actually targets of 169 among so many NFYs that are present. And we found that it is NFY2 is the most downregulated one. And if this microRNA is chelated in a target mimic like this, taking away or decoying away the microRNA, and then the NFY2A levels rise, suggesting that 169, the best target of N169D is NFY-A2. And to see its relevance in heat stress, we have generated different transgenic lines where the NFY levels are either upregulated or downregulated. This is the NFY overexpression line, and this is the resistant target where the microRNA can bind but cannot cut. This is a sponging mechanism where the microRNA is made but is sponged away by this particular construct. So basically, in all these, the target levels are high, whereas in the mutant, as well as in the 169 overexpression, the 169 levels are high and the target levels are low. So what we were able to show is that reduced level of the target is giving thermotolerance. And how how is it giving thermotolerance? So what we took up some of the known examples from literature of heat stress transcription factors and HSPs, and found that yes, indeed, NFYA2 is regulating some of them. So if it is regulating them, then does it really bind to their promoters? And we checked for all of them, and we were able to show that this is happening for HSF-A3 as well as A7B. So this is just another assay to show it in its background that indeed the promoter of HSF-A3 as well as A7B are downstream of NFYA2. And obviously, if they are downstream of this and HSF's mutant are compared with the control conditions, they show thermosensitivity. So basically, this entire story was revolving around different microRNA 169s that we identified in tomato, compared it with Arabidopsis, and showed that heat stress, HSFs get upregulated. They in turn upregulate microRNA 169s, which in turn downregulate the NFYs. And this being a negative regulator, then upregulates the HSFs, which feeds back into the particular loop. We also showed that, it's not only restricted to heat stress. We were able to show that these plants are behaving much better than wild type if it is in the drought situation, or if it even in a combined heat and drought situation. So there were other attributes that we studied, and I'll quickly go through it. What we observed was that, these plants of 169 overexpression were, bigger than the wild type. And this was true when we were seeing the entire panel, so having more dry weight. So why is this happening? So we looked at the cells of these, and what we could find was that wherever the microRNA 169 level is up or the target level is down, in other words, these cells are bigger than the wild type, and wherever the target levels are very high, these are smaller. And actually calculating them, we were able to show that the rosette size difference that we are seeing is because of difference in the cell area as well as the cell number. So this was another interesting thing for us, because what we found was that, these plants are actually having a shorter life cycle with having a shorter period for all the different growth phases. So how this was happening, we did a transcriptome, and we found that, auxin responsive genes were the most predominant class that was coming. And actually, we measured auxin in these plants, and we were able to show that, yes, the ones where the NFY levels are down, the auxin levels are high. And then which are the genes that could be potentially regulating it? We checked some of these genes which are in the auxin signaling or biogenesis or the downstream pathway. And we were able to shortlist two genes called as ARGOS. It's an auxin response gene of organ size, and ANT called ANT augmenter, which could be possibly regulating this increase in size phenotype. And we were able to show that it is only ARGOS and not ANT that actually shows the binding. That means NFY only binds to ARGOS, and ANT is acting downstream of ARGOS. And this ARGOS is actually working through a number of cells. So these mutants have a reduced cell size, have an increased cell size than the wild type. And the number of cells is basically the one that governs the structure. So we measured the yield. So please don't be bothered by so much data. I'm just giving you that, actually it has been done. So, what we found was that in the entire panel, we measured the yield parameters and everything that is written over here. And we could conclusively show that with reduced NFY levels, there is an increase in the yield. And this we also showed through the downstream target that we have found, that how it is doing it through ARGOS. So since my lab also, works on tomato, does it have any, application in a crop plant? And we repeated this in a tomato. And we were able to show that if, the target levels are down, then the plants are bigger as compared to the ones where, the microRNA has been chelated in these lines. And again, we measured the cell number and cell size, and we were able to show that in the overexpression lines, the increase in leaf size is because of an increase in the cell division. Again, like Arabidopsis, we were having that they had a shorter life cycle. And again, these are the different stages of fruiting in tomato, which was again shortened. And most importantly, we were seeing that there is an increase in the yield. And this also, to our surprise, was also happening that we had a longer shelf life. So this basically summarizes the work that we did in Arabidopsis and tomato, wherein increase 169 levels via NFY and mediated through Argos actually regulates the life cycle of the plants. I think I'll skip this because maybe, do I have time?
Not much. We may request you to please conclude, Dr. Saloni.
Yeah,
Please.
so I'll skip some of the slides.
Yeah, sure.
Yeah, and I'll just come to the LNC part because, that's part of what I started with. So I will this is just 3-4 slides and I'll go very quickly, where I'll try to show that, we in a transcription factor, which was heat responsive, we found that there is an alternative form. And both of these forms, the full length and the alternative form, are heat upregulated. And both of them actually enter the nucleus. And when we had transgenic lines, what we saw was something very interesting, because the full length were sensitive to heat stress, but the variants, they were tolerant. So looking at this particular locus, we found that there is an antisense long non-coding RNA to the entire transcription factor. And this long non-coding RNA is also heat responsive. And this long non-coding RNA is actually, if you're overexpressing it, then it is giving tolerance. So the long non-coding RNA is acting as a positive regulator. And by doing simple down-regulating the various factors like the full length and the variant and the long non-coding RNA, we were able to show that the transcription factor in its full length form acts as a negative regulator of heat stress, whereas the long non-coding RNA that it regulates and the variant acts positively. And how this is happening is because both of them actually bind to the promoter of this long non-coding RNA, and the full length has a repressor domain. Because of that, it downregulates the expression of the long non-coding RNA, whereas the variant has lost it. So let's skip this and I come to the model. So basically, this story is that we have a full length, a variant, a long non-coding RNA, which is antisense. Both of them bind to the long non-coding RNA, but in an opposite, its regulation is opposite. And currently we are trying to figure out that, how it functions. So this is basically a summary of various jobs that my laboratory has been looking at. Maybe we can discuss it some other time. And I would like to end and thank all my students who have done exceptional work and have, done the kind of work that I expect them to do and actually come up with some bright ideas themselves. So thank you very much. And I'm happy to take any questions.
Dr. Chaitra, please.
Thank you, ma'am. Thank you very much for your highly informative lecture. We shall now proceed for the question and answer session. Participants are requested to post their questions in the chat box so that we can take up one by one.
Participants, do you have any questions? Ma'am, I have one question. If, for example, one microRNA may target multiple genes.
Yes.
But maybe some gene at the specific time is very important. How it can be regulated with a specific gene at a specific time? Okay. Is there any principle behind it, thermodynamics law or anything?
Okay, so basically all the expression that we gauge is dynamic. So one has to keep in mind that while one microRNA can potentially regulate many targets,
Yes.
but the expression of the microRNA itself is under a tight regulation. It will not be expressing all the time.
Yeah, it must be
Similarly, the
controlled by
target-
transcription factor, another, yeah.
Similarly, the downstream targets themselves are not expressed continuously all the time. They are under different transcriptional or even, methylation and those kind of regulation. So what regulates is an excellent question. I think it is the activity of the microRNA, the presence of the target, and the ago with which it associates. Like I showed you that in certain tissues, it's possible if ago 1 is the most important one that leaves, but now ago 1 itself is depleted.
Then
Another layer can come that, even though you might find that transcriptionally your target was expressing, but nothing is happening post-transcriptionally.
so it is also based on the epigenetic factor and lot of things.
Right.
Okay. That's what I want to know.
MicroRNA biogenesis itself could be regulated because it's cleaved by dicers. And so dicer is also a family of proteins. So even, DCLs could be many. Some DCL are giving the larger ones, some are giving the smaller ones. And as I showed in, I tried to bring in that slide where sometimes certain agos, like in Arabidopsis and in ago 10, it is actually chelating away some microRNAs, which could potentially have gone to ago 1 for cleavage. So in that sense, it is protecting the target.
Protecting target.
So,
Another thing, alternative spliced genes, for example, they always may have premature stop codon. They may have aberrant transcript expressions. They do the regulation of its own gene, for example.
yes.
So microRNA also sometimes they can regulate their own gene.
Yes.
But do they become aberrant RNA like siRNA, or they can structurally conserved all the time?
Okay, so certain microRNA classes are highly conserved across plant kingdom, many of them. But there are some which have evolved, let's say, in a lineage specific way. Some are just monocot specific, some are just dicot specific. Recently, papers are coming where, for example, if you take rice, for example, then you can have an example where even between Japonica and Indica, the regulation of a particular microRNA is, it's just its level, its regulation, it's expressing in both of them. But an additional regulatory element has come on top of one of the varieties to bring in, let's say, chalkiness, grain chalkiness level or its flowering response.
Yeah.
So this is also true.
So they may be also degraded like siRNA. They may also become aberrant RNA to silence also sometime, I think. No?
So microRNA role is to silence a target. And there is a specific pathway for small RNA degradation. XRNs are involved in that also. Yes.
Audience, do you have any questions? So DK sir, do you have any questions?
Not a question. I do not have such deep insight like Dr. Saloni. Being a microbiologist, a bit molecular biology, I have gone through during my PhD programs and so forth in projects also, madam. I just wondering to see, have you tried your experimental approaches towards the psychrotolerant or the psychotrophic crops in cold areas?
Okay. So, I have, my focus is on heat stress.
Okay.
But so let me tell you that giving heat as well as cold stress is a little challenging because if you look at small plant, when you can still mimic those conditions. But
Right.
I want to, like I showed you data of yield and everything. I want to do that in tomato plant. Tomato is a very big plant.
Right.
We need controlled chambers going into that kind of temperature over a very long period of time, 4 to 5 months.
Right.
So one, though this is very exciting, one can actually do in seedling stages where we do check, but going right up to the an entire life cycle of a particular plant in several stress responses sometimes is challenging.
Got
So
it.
far, I have never done it in yield, but
Okay.
yes, we sometimes check our microRNAs in cold stress, but I would again say it's in the vegetative state.
Right. Do you think that another approach, if you recover the heat stress responsive gene from microorganisms that grow at high temperature,
Right.
either it may be the worthy approach if you transfer it and mix it with your plant system. So plant may survive at high temperature.
Yes, it is possible. Basically, so look, again, I would say that, there are two aspects to it. Why is a particular protein helpful? One is that, it has its internal promoter or transcriptional unit that needs to be of that kind where it will help. Other is we have HSPs, and
Right.
heat stress proteins.
Right.
They are of various kinds where they're already acting as chaperones,
Chaperones. Agreed.
or fold days,
Yeah.
where they're protecting these other peptides.
Right.
See, for example, the thermogenic bacteria and all. So they are very stable. So potentially yes, but yeah,
Yeah.
it has to be probably just engineered and look at the codon bias and put it under appropriate promoters to make it stable. Yeah.
Certainly. Yeah. Thank you, Doctor.
Thank
Thank
you,
you.
ma'am.
So nice of you.
Thank you. Very
Dr. Chetra, please lead.
Thank you very much, ma'am, for sharing your valuable insights.
Thank
On
you.
behalf of organizing committee and all the participants, I extend our heartfelt gratitude to you, ma'am.
Thank you.
Thank you, ma'am.
Thank you so much, ma'am. So, very
As
well.
we continue with the session, it is my immense pleasure to introduce our next prominent speaker, Dr. Prashant Vikram. Dr. Prashant Vikram is an accomplished agriculture scientist, and plant breeder with an extensive expert in plant genomics, molecular breeding, and crop improvement. He has built a distinguished research career through his association with leading international and national agriculture research organizations, including the International Rice Research Institution, International Maize and Wheat Improvement Center, International Potato Center, and several in ICAR institutions. Dr. Vikram earned his PhD from IRRI Philippines, where he also pursued postdoctoral research in advanced crop genetics and molecular breeding. His research expertise includes wheat improvement, genetic transformation, stress tolerance, and genomics assistance, breeding for climate resilient agriculture. Through his innovative research, he has significantly contributed to the development of the improved crop varieties capable of withstanding environmental stress while enhancing the productivity and sustainability. An active researcher and a scientific contributor, Dr. Vikram has published more than 50 research articles in reputed international journals and has served in leadership positions, including a senior scientist and a director of research in agricultural sciences. His recent academic and research engagement includes the association with the international agricultural organizations, ICR institutions, and SGT University, where he contributed to the strengthening agricultural research and innovation programs. Dr. Vikram has received the several prestigious recognitions, including a Jawaharlal Nehru Best Thesis Award from ICAR and Ramanujan Fellowship, etc. With a strong interdisciplinary approach integrating plant biology, genomic, molecular biology, and stress management, Dr. Prashant Vikram continued to contribute towards the sustainable crop improvement and global food security under the changing climatic conditions. I now invite Dr. Prashant Vikram to deliver his lecture and share his valuable insights with us. Sir, over to you.
Sir, yeah, please join. I think, yeah, you can join.
Well, I have joined you.
Your video. Yeah, yes, you can upload.
Okay. So good afternoon, everyone. Can you see my slides?
Yes, we are seeing, sir. Yeah, you can go ahead. Good afternoon, sir.
Okay. So thank you, Karthik, for inviting me for this talk. Uh, as the title mentions, I will like to share my experience in ERI and CIMIT on the Green Revolution, my experience with the Green Revolution genes and their application in the breeding and pre-breeding programs. So, as the title mentions, looking beyond or behind the Green Revolution. So, we are all well aware that that now is the climate change era. So I would like to present this slide and just go through the three pictures. There are 1, 2, and 3. One is the era of like pre-green revolution, when there were tall varieties in the harsh environment, and low yielding germplasm was there. So although population was less, but still that was insufficient yields were there. Then came semi-dwarfing rice and wheat varieties, which brought sort of monocropping. But these were input responsive genes were introduced, the SD1 gene in rice and RST genes in wheat. And then the yields became we India and Pakistan, we became exporter, rather we were importer in the pre-green revolution era. But now after 2000, when entering to the 21st century, so the climate change impact is visible. Now it is worsening year by year. So again the debate came, what we need actually in terms of genetic gain, what kind of genetic makeup we need for rice and wheat varietal germplasm. So we actually need we talk a lot about gene bank use and pre-breeding. Now the whole world is after gene bank use and pre-breeding, just to cope up with the climate change era. What I want to convey here is the new thing is what I learned from rice and from my 12-year experience in CG centers is we will have to shift the threshold. Right, when I say threshold, it means if you will increase the stress tolerance, you will have to compromise in terms of yield. Right, if you will reduce the crop cycle, then there is compromise on yield. But at the same time, if you try to shift the threshold, when I say shifting threshold, it means 1 is to 2 is equal to 2 is to 4, and that is equal to 4 is to 8. Right, which means there was a capping of 100 centimeter varieties. The breeders in rice and wheat, they both wanted to have not more than 100 centimeters to avoid lodging. But whatever threshold is now, is in that threshold, this 100 centimeter capping is fixed. But if you increase this 100 centimeter to 110 centimeter, let's say 10%, if you increase, then we have a scope to play around with the flowering duration and yield. Right, so this is one way to play around in the to achieve the genetic gain through pre-breeding and speed breeding and shuttle breeding. Now I will explain what I learned in rice and what I learned in wheat, and then high density genomics used in this process, and then the impact pathway. There are four in the four sections I will discuss. Coming to rice, we identified a large effect QTL. Actually, it was my PhD work when I identified this large impact, one QTL showing effect simultaneously in multiple populations, QDTY 1.1. So this is the one of the most popular QTL ever used in drought, for yield under drought in rice, and marker-rested breeding varieties have been developed using this QTL. But the issue with this QTL was that this QTL was very close to the SD1 gene. Now, when we looked around the SD1 gene, what we found that there were multiple genes associated with SD1, and they were wiped out in the due course of development of green revolution varieties. And then you can see here there are two figures: pre-green revolution land races, in which you have the SD1 and on the both sides of the M1 and M2 are the flanking markers of the QTL region. So T corresponds to tall, and N also corresponds to tall, because the N here is N22, which is the tall parent, and N corresponds to the wild allele of SD1 gene, and then T again corresponds to the wild allele of the parent, that is N22. So this T and T means all the tall are here, which is coming from the wild parent. So it was there in the pre-green revolution land races. This situation was there, but in post-green revolution varieties, what happened when they were crossed with the IR8 or as SD1 donors, all this region was shifted. This change along with SD1 gene on the adjacent area also was replaced by the dwarfing from IR8. As the result of which, what happened? Those genes and alleles which were linked with SD1 for abiotic and biotic stress tolerance, they were lost in our modern day varieties. So then we realized from when I started looking after it, you see you see this green and yellow picture. These green ones are the pre-green revolution rice land races, right? So in these land races, you can see the whole region is N. N correspond to N22 type, means tall type, means wild type, and S correspond to the Swarna type, that is what coming from IR8 or IR64. So, but what we actually need that dwarfing allele of SD1 gene, but on the both side of SD1 gene, we need wild, the genomic region from wild, that is the tall parents. That's why it is here written, it is tall. So on the left side, you have, see, we need desired combination. So it took us our to break this linkage, it took us around 10 seasons. So I will show you the series of efforts what we make to break the unholy linkage between drought QTL. The QTL markers are here, M1 and M2, and the SD1 gene, which is again in the center of that QTL region. See here, this one is the gene, and M1 and M2 is on the both sides, right? So it took us around 10 seasons to break this linkage. So we generated first of all 3000 plus BC2F2 plants, and then we took out the dwarf plants, and then we genotyped. We found 2 plants segregated for the QTL region as well as SD1 gene, but no, not SD1 gene, but the dwarf parent allele of SD1 was there because the plants were dwarf. Later on, we confirmed, and then the plants died. So then we retuned and split planted. We then again allowed lines to segregate, and then we fine mapped the DTY and 1.1 QTL, and then we broke the linkage simultaneously, and we developed rice genotypes which were dwarf but drought tolerant. Now, what was the outcome? You can see 115 days after sowing. Here, Swarna, the recipient parent, the green revolution variety, you can say, this did not flower well, but at the adjacent, you can see the nil, the near isogenic line developed from the cross of Nagina 22, the tall line, the pre-green revolution line, with Swarna. So this near isogenic line is flowering very well. 115 days after sowing under drought stress situation. On the same date, you can see you cannot find difference between Swarna and the near isogenic line. So this was the outcome of our this 10 season effort to after breaking the linkage between the SD1 gene, this gene, and the genomic region on adjacent side of this gene. We have used we have developed a multiple stress tolerance variety using this using this donor, this IR 91659-54-35. This was the line number, and this we developed multiple stress tolerance variety CR Dhan 801, and it covered more than 10,000 hectare in 2019. So this was the whole outcome of our effort, and the donor which I mentioned, the dwarf drought tolerant donor, was used to develop multiple varieties. Even it is even today, breeders and plant geneticists are working on it throughout the India and Asia to develop the next generation multiple stress tolerance variety. One example here is CR 801, which has a drought tolerance yield QTL as well as submergence QTL, and it was released for the lowland and for the irrigated ecology of rice wheat Indo-Gangetic plains and other areas as well. So this was all about the rice, our rice study and the line development for next generation rice varieties. Now coming to wheat. So wheat is a little bit different than rice in terms of genetic base, because the origin of Oryza sativa is said to be like Indochina, which is a confined region. But on the other hand, if we talk about wheat, it is still not clear that which part of Fertile Crescent is the center of origin, right? Like Syria, Lebanon, Iraq, Israel, Jordan, Iran. See, wherever this war is going on, the whole region is corresponding to the center of diversity for wheat. But at the same time, we have to admit that in wheat there is enormous genetic diversity. We have, we have tetraploid wheats, and we have hexaploid. In tetraploid, we have dicocoms, we have dicocoms, we have immer, then we have durum, and in hexaploid also we have we have estivum. Within estivum, this B genome is not known, not very well characterized. But also there with respect to the grain type, there is hard red winter wheat, then amber wheat, then white wheat. Then there is winter wheat in Europe, and in northern hemisphere winter wheat is common, which is grown for which the crop cycle is almost 6 months. And the spring wheat which we grow usually here is around 130 days, 135 days. So there is enormous genetic diversity. And one more thing with wheat genome is that wheat genome has a very high buffering capacity. Even the buffering capacity of the wheat genome is so high that it has digested one chromosome R from another species, Sickle cereal rye, 1R1S translocation. See, the one chromosome R is from Sickle cereal, and wheat is still wheat. So you will not find this kind of buffering capacities and in any other crop. That's why wheat genome study is entirely different. So for the in the wheat green revolution varieties were also like rice. They had from Norin 10 they had RST1 and 2, and they brought monocropping, and then the genetic base was narrowed, but not as narrowing like in rice, because wheat genome is vast, is around 35 times more bigger than the rice. So that's why the negative impact of green revolution on wheat is not as on the rice. This is one important point and aspect. But yes, the genetic base was narrowed because the breeders and that narrowing was not only because of RST gene. That narrowing was because of rust problem. So breeders don't want lines which are having the susceptible alleles for rust. So that is why that was one of the reason why we have narrowing genetic base issue in wheat. So for the next generation, for the climate change era, we need to have varieties which that can cope up with climate change, and for that we need to have varieties with larger genetic base, right? So we took Iranian land races from USDA, which was originally collected from Iran, from the Fertile Crescent of Iran during the Iran-Iraq war. As you can see, this whole series of events collected by Dr. Qualset in UC Davis, and then given to USDA from USD from in CIMMYT. We did GBS analysis to genetically characterize, and and at the end we could not establish this genetic diversity with geographical diversity. And there were two possible reasons. One was that since the collection was not very appropriately done, because it was done in the Haribari period, in the war period by Professor Mansoor, and it was given loosely to Dr. Kwalset. So that might be one reason, and the other is since Iran is one of the center of origin, so there might be fast movement of germplasm within Iran. So that these two might be reasons why we did not establish the relationship between genetic and geographical diversities. So we have reported that in the paper, and we further we made core set using genotype and phenotype matrix. We merged both matrix into one, and then we calculated Gauss distance. We made the group using that distance matrix, and then from the groups we took sample as core set, and we used core set for the pre-breeding line development. And same we did for Mexican Creoles. Now you will say, what is Mexican Creoles? What Mexico has to do with the wheat? Actually, if you go to the history of Latin America, around 500 years back, Spanish conquered this Maya and Azteca civilization when they entered to Puebla port and other ports in Latin America. So they brought wheat from the Spaniards brought wheat, and then they introduced, which survived very well in different parts of Mexico, and then they were later on they were called Mexican Creoles, right? So we did Dr. Borlaug and Dr. Scovement actually collected those Mexican land races, those Creoles, and then when I was there, we genotyped with the GBS, and then we made the groups, but the groups were unlike the Iranian case. In Mexican land races, the genetic diversity corresponded to the geographical diversity very well. That's why we were able to show these groups in the map of Mexico. This is not just arbitrary. This map is made by this ArcGIS software, and then everything is put there through that software. So it was done very well, and then with the same strategy I mentioned before, we made the core set, and then the core set were evaluated for disease abiotic stress and biotic stress, as well as quality, and then selected lines were used in pre-bidding, and these lines are still being used in different part of world, the core sets and the pre-bidding lines. I will mention later on. Now in the fourth section. Now coming to the high density genomics data and tools, how they are why they are important for gene bank use. So some of the examples I would like to explain. As I mentioned, in case of Mexican Creoles, we analyzed and we found that with the help of this high density genomics data, means from 8,000 line and 20,000 markers. So we found some rare alleles in the which were found in the land races from Michoacan. Now you will say, why it is Michoacan? See, if you divide Mexico into half, the upper half is hot and dry, and the lower half is cold and wet. But in between the region, in the borderline, you will find a state, Michoacan. So in Michoacan is unfortunately it is famous for drug cartels, but at the same time it has a peculiar climate. So there, hot and humid climate is there, and it is hotspot for wheat diseases, not the rust, but other wheat diseases like Fusarium head blight. So in this environment, for the land races which were specific to this region, they were having rare alleles, and probably these. And then we later found that these alleles are for associated with the resistance to some specific diseases. So this was one example with what we found while studying the land races. And as I mentioned, we did pre-breeding. So in case of wheat, as I mentioned, the wheat genome is having high buffering capacity. So we should not expect that we make crosses of land race or exotic with the cultivated and we will get the desired combination. No, it is not possible, because of the buffering, because the wheat genome is very big, and just by one combination, one cross, we cannot get the desired combination. So what we did, we made three-way crosses, not top cross. We made three-way crosses, means exotic into elite, and then the F1 was crossed again with another elite, and then the TC1 F1 which came, three-way cross F1 which came. that we advanced, and then we made selections using that. So these exotics were actually the core sets what we identified, what we presented before. And then see, if we in the three-way process, if we advance the TC1F1, the expected contribution of exotic was less than 25, because A cross B and F1 is 50%, and if you cross F1 again means here B means elite, so it will be 25%. And then if we select based on elite character more, so further it should go below 25. So what we identified that while doing GBS analysis of pre-breeding lines, we found the exotic contribution was less than 25, which was expected as what we did practically in the field. Then we identified that one of the excellent line in CIMMYT, which is used for developing multiple wheat varieties in India and different parts of world, called Kachu. Why it is so? It has a broad spectrum of resistance, rust resistance. So we analyzed and in the real population, and we found that it has a segment from 2NS segment from Aegilops ventigrosa. So again, it is like it is coming from wild to this cultivated line. So that makes this line broadly resistant to against the different rust strains. Then for heat tolerance also, in pre-breeding we identified that there was a heat QTL on chromosome 6D, and we found that the haplotype which was responsible, we further studied that in the haplotype from where the resistance allele is coming. So again, it was Aegilops toshii. So it means the heat tolerance was contributed from the wild here again. Then there was a theory that land races don't have rust diseases. That's why breeders don't prefer it. So, but when we did analysis, practical analysis, then we found that no, it is not true. Land races do have rust resistance genes, but yes, not very much resistant. But there are some degree of resistance to land races have, which gives the message that land races should be used for for the crop improvement purposes. Then coming to rice, in one of the recent paper, we have seen that we have found that from transcriptome-wide genome analysis, the markers for low GI have been identified. See, low GI is a trait which is bottleneck for breeders, because to analyze low GI, we need to have a set of people who can donate their blood. So for that we need multiple regulatory approvals. So that's why see, clinical analysis of GI in human being is not really an easy task. So that's why, but there was somehow in ERI, the one in the NESE group in ERI, they managed to have it, and then they published that two genes, one on chromosome 6 and on chromosome 9 in rice, which were associated with low GI using transcriptome-wide association analysis. And these markers are being used to develop the low GI rice varieties, which is the demand for like for today and tomorrow as well. So these were some examples how high density genomics data have been used for the practical pre-breeding and next generation climate smart nutritive varietal improvement in rice and wheat in our ERI and CIMMYT programs. Now coming to another pathway, another important point is gene bank really important for the impact pathway. So I will like to mention some fact that Norin 10, the Norin 10 line, which was developed by an American from a landrace Daruma, it contributed RST1 and 2 in wheat. And similarly, one landrace, Japanese landrace Akako Mugi, contributed RST8. So and also in case of rice, SD1 gene is from landrace DGO Ugen. So apart from that, for multiple physiological traits like cold, drought, heat, height, multiple ovaries, salinity, and other traits, the alleles are coming from genetic resources. See, as you can see in this table. And what I have seen with my own eyes and my own study in CIMIT, I found one big spike, like, and then I tried to see how come the grains are so big. So then I found that you can see on the right side, see this large pistil is here. The gene bank line is having, and then the green revolution variety PBW343 you might have heard. See, the pistil is so small, and then the recent CIMIT line, which is very popular, it is also having small pistil. So if we somehow transfer this large pistil trait from gene bank to the cultivated ones, we can get some increase in the yield. So gene banks are really important, but we have to strategize their use in the breeding programs so that we can have a speedy recovery of recipient genome with added multiple alleles from the gene bank lines for stress tolerance and quality and yield as well. Then, in case of wheat, see, in the we need see, the wish list is so high. So to achieve all this semi-dwarfness, high harvest index, tiller number, long stem, large spike, large grain, fertilizer use efficiency, large root system, to achieve all this straight into one is not an easy task. So for that, we will need shuttle breeding, speed breeding, and genomics assisted breeding to achieve it in a precise manner and in a popular backgrounds. See, what we did practically, what I told you, just it is not so. It was not so easy. This was our flow chart, like how we started and how we performed all this. We have delivered it. See. Again, so I'm skipping that slide because I will show you the impact later on. See, one thing is very important I would like to mention in the crop improvement process or breeding improvement breeding. One thing is very important. We cannot compromise with the number game. The more we have number in the field, the more we will having the chance to get the desired plant type, because accumulating different alleles into one is not an it's not a 1 season or 2 season task. So that's why always we should prefer a large number in the field to if we want to have multiple traits into one. Then we have developed a series of pre-breeding lines and landrace core set, and we have shared it with the different groups in the world. You can see in India, Iran, Pakistan, Iraq, Turkey, Icada, Lebanon, Portugal, Spain, UK, and different places in USA, and then in the Embrapa, Brazil and Canada. So in Australia also we shared. So we have shared our germplasm, and then they are really using it in their wheat development program. But this is one example. So this is one example, and this is a pipeline. This is the product pipeline, and this pipeline should continue if we want to have genetic gain. So this is what we learned from our pre-breeding program. And last but not the least, we can do our effort, but we cannot control everything. The guy who identified the green revolution gene in rice, look what he said. Although we do not know the absolute origin of GGO gen, we assume the original variety was brought to Taiwan from mainland China before Japanese occupation. There is no clue whether the short stature mutation occurred in China or Taiwan. So China and Taiwan, they are arguing that the SD1 gene belongs to me and you. Like, apart from semiconductors, they are fighting. They are arguing for this SD1 gene also. That right. So nobody knows from where it came. So this is one. Then there is a popular variety Swarna. It was very popular before it was released. Again, one of the most popular variety in Sonora Valley of Mexico. It was just a line of a student, right? And it was of a QTL mapping population, Sibyl Herrera. So Cirno was identified, and then it was later on it was released as a variety. Similarly, one of my varieties, CR 205, it was just a line for a mapping population, but it was released as a aerobic rice variety. So we can make our effort. We should not compromise with the number game. We should not compromise with the. We should use all sort of effort to have a precise and fast track recovery of the genome, and then add as much multiple genes as we can. So this is all what I learned, to have a next generation climate smart nutritive varieties. And finally, I'll repeat the same slide. We need stress tolerance genes. We need quality genes from pre-green revolution era varieties and the yield genes from the post-green revolution varieties, and to have a next generation varieties well suited for the climate change era. So with all this, I would like to acknowledge the drivers of green revolution, late Dr. Sanjay Rajaram and Dr. G.S. Kush. Both are World Food Prize winners, and all the researchers who have contributed directly or indirectly in my career journey. With this, I would like to say thank you, and questions if you have, please. Thank you very much.
Thank you so much, sir, for your insightful lecture. Now it's open for discussion.
Audience.
Participants, if you have any questions, you can post the questions in the chat box.
Audience, do you have any questions? Sir, I have one question. Iranian land races, I know you are using long-term Iranian land races.
Yeah.
Compared to Indian land races and other land races, what kind of specificity you found with that? Because I also did some work with you. In Iranian land race, when we were doing, we found some less diversity. Maybe wheat is generally autogamous. So maybe we can expect generally less diversity compared to allogamous species. So how it can be used in the future, Iranian land races, kind of specific land races, to improve the yield and other attributes?
Okay. See, you might be aware about facultative wheat. You know, there's winter wheat and spring wheat, and in between there is facultative group.
Facultative.
Right.
Yeah. Mm-hmm.
So Iranian land races are see, their facultative is not just one class. You cannot classify something as facultative. It means the facultative may go more towards spring wheat, and it may go more towards the winter wheat. Right? It may flower in 140 days. It may flower in 150 days. So why you are saying it is less diverse? Because you have grown this facultative type in the spring seasons of Mexico or India, right? If you see, if somebody put you in Alaska, so you will not behave normally. Same thing happening with the Iranian land races. So their home is in Iran, so their environment is different, right? So they belong to more and more northern hemisphere. See, some of the Iranian land races may flower. They do flower, not all of them. But if you grow Sardari land races from Iran, so they don't flower at all. They flower only after 160, 170 days. Because their facultative character is more towards the winter wheat. So this is the answer of half of your question.
Yes,
why you don't observe diversity, and the second is how we can use it. What we can use, these Iranian land races have an excellent grain filling property. They flower very late, but the grain filling is very fast.
So yield will be we can get more yield.
You please get my point. They flower very late, but the grain filling is very fast. That too under heat stress condition. So if you can pull out that gene or genomic region associated with grain filling, and you put it into spring, you may get a genetic gain. But for that, you will need to do marker assisted backcrossing or some other approach. Right? You cannot do it with simple breeding. So that's what I mentioned. So you need to club, speed breeding, shuttle breeding, and high density genomic approach, and gene bank use as well.
Yeah, nowadays even genome editing, we can save the time sometime doing.
But in case of wheat, do you think you can achieve something?
But some extent it is possible, not like rice, but being a polyploid, we can try.
Some large effect genes like rust or height, you can do that. But for quantitative traits, it's not possible.
Okay. Sir, DK sir, you have any questions?
Yeah, no questions, but sir, sharing of knowledge a bit. You discussed about rice variety with low GI. And as per
Yes.
my knowledge, there are several varieties available in India. We can purchase also at home, like sugar watchers, if I'm correct.
Yes, yes.
Guy Bismart, 24 Mantra Organic, etc., and so forth. So these varieties are applicable if we consume these at homes, which may permit us not to solubilize starch. Do they have more starch content in the rice? So starch should not be solubilized in a quick manner upon consumptions. So what is your opinion, what your prospective futuristic research to reduce the level of the sugar or starch in the rice? By which way or which method shall you deploy?
See, what they are selling in the market in name of parboiled rice. What rice you eat in the is polished rice actually.
Okay.
In rice, you have to remove when you remove this yellow color. If you see in the picture, this yellow color is husk. When you remove this husk, you don't have this white one.
Right.
You have, there is another layer, bran. So if what most of the companies are selling, they are selling rice with the bran, and they call it a diabetic free rice. It
Right.
is not like that,
Okay.
right? Right. So
Right.
there is no variety so far which is available, which is developed by breeder, and that too for low GI. So first, that's why I want to clarify what in wheat also they are selling diabetic wheat, which is actually not diabetic wheat, which is immer wheat, the tetraploid wheat, right? So it is good for it releases sugar very slow. That is true. And if you eat parboiled rice, which they call it diabetic rice, parboiled rice is very common practice in Eastern India, and in our ancient days also it was very common. But nowadays we like polished rice, so we remove the bran from the rice and we eat it. That's why it is having the high soluble starch.
Okay.
Right. So that what I discussed to have low GI is to having low GI in the endosperm. But if you want to avoid, you can simply eat parboiled rice. You can just type parboiled rice, that is having high vitamin B, and that is having low GI as well. Yeah.
Okay. Okay. Thank you. Thank you, Doctor.
Welcome.
Thank you. Chatra,
Welcome.
please.
Yes, sir. Thank you, sir, for your valuable lecture. And as we
Welcome.
conclude today's session, it is evident that the recent advancements in genetics and genome engineering are transforming the future of agriculture. Modern tools such as marker-assisted breeding, genetic genomic selection, and genome editing technologies have been opened new possibilities for developing a climate resilient crop varieties with enhanced productivity, resource use, and efficiency, and resistance to a biotic and abiotic stresses. In the face of climate change, growing food demand, and shrinking natural resources, these innovations offer a powerful solution for achieving a sustainable and climate smart agriculture system. However, the successful adoption of these technologies will require a continuous research, supportive policies, and ethical consideration, and effective collaboration among the scientists, policymakers, industry stakeholders, and farmers. I sincerely thank both the speakers of second session, Dr. Prashant Vikram and Dr. Saloni Mathur, for sharing their valuable knowledge and insights, and all the participants for their active engagement throughout this session. And I hope the discussion and learning from this program will inspire further research, innovation, and application of genetic and genomic technologies for building a resilient and sustainable agriculture. Thank you all for your presence and participation. We look forward to welcome you again tomorrow. Thank you.
Thank you, sir.
everyone.
Thank you, sir. And Chatra,
Thank you also.
please invite others for tomorrow's session as well. Thank you.